Hey guys, welcome to Human A&P1. My name is Trevor Lohman. This is my 6th or 7th try trying to record audio to a PowerPoint, so bear with me. I'm just going to try to make it through, even if I make some mistakes. Our first chapter is Chapter 3, the cell. Cells are the living units. They're the smallest living units. We can't break the body down into smaller pieces that are functional and living. This ties in a bit to cell theory. Cell theory is a theory that describes cells, what they are, how they fit into the body, and the theory kind of gives us some context when we talk about cells and think about their function. There's 4 principles to cell theory. The first is that a cell is a basic structural and functional unit of living organisms. We touched on this a bit. This principle is just saying that it's the smallest building block that's living in the body. There are smaller building blocks like molecules, atoms, things like that, but they're not living, and they don't come together functionally. They don't have a common function like cells do. Cells are individual units that are living and have a purpose, have a function. All right, second principle. The activity of an organism depends on both the individual and the collective activities of its cells. So the activity of one cell can affect the organism, as well as the activities of many cells can dictate the activity of an organism. So if a bunch of cardiac muscle cells come together, they can form the heart and be an important part of circulation. Same with other organs and tissues of the body. Okay. Principle 3 is the principle of complementarity of structure and function. This is a fancy way of saying that structure equals function. Let's look at a muscle cell versus a fat cell. So a muscle cell, its structure allows it to contract and shorten. If it wasn't for that structure, muscles wouldn't be able to contract and shorten and create movement. Whereas a fat cell is designed to store adipose. So based on their structures, those two types of cells will have different functions. So structure equals function. Last principle here is that the continuity of life from one generation to another has a cellular basis. So every cell contains a copy of our genetic material, and that material is passed down every time a cell divides. So that keeps our genetic code continuous in our body. We pass down half of our genetic code, or any living organism passes on its genetic code through cells. Okay. The last part here in this slide, and this has to do with the image here, is the idea of a generalized or composite cell. So when you see pictures of cells in books and on the internet that aren't a specific type of cell, they're probably this composite cell, which is basically a drawing that includes all the different components that most cells share. So there probably isn't a cell in the body that looks just like this. This is just giving you an idea of the structures and subunits that most cells have. If we really zoom out and look at a big picture of cells, the 3 main components of a cell are the plasma membrane, the cytoplasm, and the nucleus. Not all cells have these. For example, the red blood cells don't have a nucleus, they're anucleate. But in general, most cells in the body share these 3 general components. All right, let's start with the plasma membrane. So the plasma membrane is pretty obvious what it does when you look at a picture of a cell. It creates the boundaries of the cell. It is the exoskeleton. It contains all the fluid within the cell, and it separates that fluid from the fluid outside of the cell. If there wasn't some type of container, the cell would just sort of dissolve and diffuse. It's kind of similar to an egg, where if the yolk's a nucleus and the egg white is the cytoplasm, then the shell is the plasma membrane. It contains the egg. When we talk about the plasma membrane, or when we look at a model of the plasma membrane, you'll hear it called the fluid mosaic model. This kind of paints a picture of how the plasma membrane works. It's exceedingly thin. In the book here, it says 7 to 10 nanometers thick. It's composed of a double layer or bilayer of lipid molecules with protein molecules plugged into or dispersed in it. The proteins, many of which float in the fluid lipid bilayer, forming a constantly changing mosaic pattern. And that's where the model gets its name. I just read that bit from the book there. This idea of a lipid bilayer is really important. Because the plasma membrane is set up as a lipid bilayer, it allows cells to do many of the things they do. So if you look at this picture, we have little pink spheres with tealish tails coming off them. Each one of those is a phospholipid. Phospholipids make up by far the vast majority of the cell membrane. What's important about phospholipids is that their phosphate head, the pink sphere, is polar. So if you think back to chemistry or biology, when something's polar, it means it has a charge gradient on it, meaning it will be attracted to other polar things, because within it, it has a positive and a negative charge, and like a magnet, the positive and negative charges are attracted to the opposite charges. So, one example of something that's polar is water. And most of our cells, both our cell and our body's fluids, is water. So the phosphate polar heads tend to point outwards, out of the cell, towards the water on the outside of the cell, and inwards on the water inside the cell. So that's why when you look at this picture, the exterior and interior of the plasma membrane is made up of phosphate heads. The interior is made up of lipid tails. Lipids aren't polar, they're nonpolar, so they're attracted to nonpolar things. Another way to think of it is they're nonpolar, so they repel polar things. They repel water, and they're attracted to each other. That's why the lipid tails tend to be attracted to each other, and that gives us the arrangement that we see in the picture. Polar heads, phosphate heads pointing away towards water, and lipid tails pointing towards each other. So there's no glue or structural anchors that need to hold the plasma membrane together. It's just electrical forces and the nonpolar attraction of the fatty tails. So this, you guys probably know this already, but when something's polar, it's said to be hydrophilic, which means water-loving, so the polar heads are attracted to water. And when something's nonpolar, it's said to be hydrophobic, so it doesn't like water, it's afraid of water. Typically, lipids and fats are hydrophobic, and polar structures are hydrophilic. That's just another way of thinking about the forces that cause this membrane to assemble like this. Okay, there's also glycolipids. These are fats with sugar chains attached. These are part of something we'll talk about later, the glycocalyx, or calyx, and it just serves in cellular identification. We'll get into that later. There's also cholesterol. Cholesterol is a little yellow bunches of fat or of cholesterol inside the plasma membrane that you can see in the picture. Cholesterol adds rigidity to the membrane. It gives it some extra structure. Lipid rafts. Lipid rafts I'm going to read right out of the book. Here we go. About 20% of the outer membrane surface contains lipid rafts, dynamic assemblies of saturated phospholipids, associated with unique lipids called sphingolipids. You don't need to know that. And lots of cholesterol. The quill-like lipid rafts are more stable and less fluid than the rest of the membrane. That makes sense, right? Because I just told you that cholesterol is rigid. So these lipid rafts that are mostly cholesterol are very rigid and stiff parts of the membrane. Lipid rafts are assumed to be concentrating platforms for certain receptor molecules or protein molecules needed for cell signaling. And this is important when for cellular invagination, like endocytosis, which we'll talk about later. That's when the membrane takes in contents from outside of the cell. But for right now, for this slide, I just want you to know that there's these four different... Okay. So there's three different general categories of proteins in the plasma membrane. The first is integral or integral plasma membrane proteins. These are proteins that span the membrane. So they span the cell membrane interior, that lipid interior, they cross it from one side to the other. A lot of times these proteins are going to be pumps, things that pump certain structures that can't pass through the membrane across the membrane. That's something else I wanted to talk to you about that I forgot earlier. and that is that the structure of the plasma membrane, because it has a polar exterior and polar interior, it's difficult for fatty things to pass through. However, let me start that over. Because of the structure of the membrane, it's difficult for polar things to pass through because it's difficult for them to pass through the interior. That non-polar interior is difficult for things with charge to pass through the hydrophilic lipid interior. Things like water can enter the membrane through channels specifically for water, but other than that, it's difficult for polar things to pass through. Things like steroids or other lipid-based compounds, fat-based compounds, can pass through because of momentary openings in the phosphate heads. And then the lipid molecules, they have no problem going through the lipid interior. And then those momentary openings on either side of the membrane allow them to slip in and out pretty easily. So if you have a drug that's fat-based, like a steroid drug, it passes right through the membrane without a pump. But I'm kind of jumping ahead of myself here. For now, I want you to know that the lipid membrane structure, its polar and nonpolar structure, kind of plays a role in what things can get in and out of the cell without help. Okay, back to proteins. Sorry. Integral proteins, those span the membrane, a lot of times are pumps. Peripheral proteins, those are proteins on either the interior or exterior of the plasma membrane, and they don't span that lipid interior of the membrane. And then the glycocalyx. So these are proteins with attached sugar molecules. So we talked about glycolipids. Those were fats with attached sugar molecules. The glycocalyx is proteins with attached sugar molecules. Those glycolipids are also part of the glycocalyx, so it gets confusing. But basically, the glycocalyx is just the sugary coating our cells have, kind of like an M&M. So the sugary coatings allow other cells to identify whether or not that cell belongs. Every cell has a unique glycocalyx. It's like a fingerprint that allows other cells to recognize it. It also plays a role in structure of tissues because that sugary sticky coating kind of allows cells to adhese themselves to each other a bit easier. It's almost like sticky glue. And a good picture in the book to see all this stuff is figure 3.3 and 3.4. All right, let's talk about cell junction. Okay, 3 basic types of cell junctions. The first are tight junctions. Now, these are a lot like they're described. They're junctions that hold cells together really tightly, so tightly that molecules cannot pass between these junctions. So you're going to see tight junctions anywhere where you don't want chemicals, enzymes moving down through the cell layers. So in the cells lining your stomach, there will be a lot of tight junctions because you don't want that stomach acid leaching down through the cell layers into the basal, the lower layers of cells. We have desmosomes. These are really strong cell junctions. They're almost completely structural in purpose. They're a lot like rivets. So if you look at the picture in the top left, it's a lot like a little rivet with actin filaments extending out into the cytoplasm, out into all the fluid inside of the cell. These allow for really strong junctions between cells. You'll see these in places where there's a lot of shear forces, like the skin. And then lastly, we have gap junctions. Again, the name's kind of intuitive. They're junctions with tunnels. So they hold 2 cells together and provide a passageway between one cell to the other, the fluid from one cell to the fluid of another. This is important in cells where chemical and electrical communication is a must, like the heart cells, for example, the cardiac cells. You want that electrical wave to pass over the entire heart, so the whole heart beats. You don't just want individual pieces of the heart to beat. So you'll see those in electrically excitable tissue, such as cardiac muscle and others. All right, let's talk about membrane transport. All right, guys, this is kind of a tough slide. I just recorded like a 20-minute thing, and I had to hit reset. So There's something I realized I forgot to talk about earlier, and that has to do with the plasma membrane. Inside of the plasma membrane, the fluid inside of the plasma membrane is called the intracellular fluid. Makes sense, right? The fluid outside of the plasma membrane is called the extracellular fluid. It's also called the interstitial fluid, or the fluid between cells. This fluid is derived from blood. It's not blood, it's the fluid component of blood that contains solute, like salts, potassium, proteins, carbohydrates, minerals, all different types of things. And it's from this interstitial fluid that cells bring in things they need and get rid of things they don't need. And that's what we're going to be talking about, membrane transport across the membrane. And here's what I forgot to tell you. I'm just going to read from the book. Okay? Although there is continuous traffic across the plasma membrane, it is selectively or differentially permeable. It allows some substances to pass while excluding others. We talked about that a bit. It will allow fat-based compounds in or out freely, but not polar and large polar molecules. It allows nutrients to enter the cell, but keeps many undesirable substances out. At the same time, it keeps valuable cell proteins and other substances in the cell, but allows waste to exit. Okay. Substances move through plasma membrane in essentially two ways, passively or actively. In passive processes, substances cross the membrane without an energy input from the cell. In active processes, the cell provides metabolic energy, usually ATP, needed to move substances across the membrane. So in order to get the things out of the cell that don't naturally just passively go across the membrane on their own, like lipids, it needs to actively export them. Or say, for example, these lipid or some other type of protein molecule or water, or actually not water, or some nutrient has a high concentration outside of the cell and is able to cross the cell membrane into the cell, you need a way to get rid of it, to export it, to keep the concentrations where you want it. So transport across the membrane, just to back up, can occur passively or actively. So let's talk about passive processes first. The first type is diffusion. I would put osmosis first, but I want to talk about diffusion because osmosis is a type of diffusion. Diffusion, okay, if you put something in water, say you put a salt tablet in water, the sodium and the chloride in the salt tablet will diffuse through the water. If you leave it in there long enough, the movement of the molecules colliding will eventually cause that tablet to dissolve and spread throughout the water. Okay? That happens because solute moves from areas of high concentration to areas of low concentration. So when you drop that salt tablet in there, there is a 100% concentration of salt at the tablet and a very low concentration of salt throughout the water. So it dissolves and diffuses throughout the water to help equalize the concentration gradient. Okay, now that I've described diffusion, I'm going to go into osmosis. So osmosis is diffusion of a solvent, usually water when we're talking about body. So the example I just gave of the salt was diffusion of solute, which was the salt, diffusing throughout the solvent, which was water. The same thing can happen in reverse in the example of osmosis. So if you have an area where there's a lot of dissolved minerals, solute, say for instance in a cell, and those can't get out, either because the cell doesn't want them to get out or they can't cross the membrane because they're polar, then water can come into the cell to lower the concentration gradient. So both solvent and solutes naturally want to equalize a concentration gradient across a membrane, across a permeable membrane. So if the salt can't get out of the cell, the water will come in. And that occurs through channels, protein channels, called aquaporins. Water can flow in and out of the cell. And it is the solute and solvent relative concentrations on either side of the membrane that determine whether water is flowing into the cell or water is leaving the cell. And these concentrations are called osmolarity. Okay. we're going to skip over hydrostatic and osmotic pressure for a second and talk about tonicity. So let's look at a cell. Actually, no. Let's talk about using an analogy first. So when you go swimming, you get pruning. Okay, that's because you're swimming in a hypertonic solution. What that means is that the concentration of solute is higher in the pool water than in your own body's water. So the water in your body is more pure. It's more pure water. It has less solute in it than the pool water. Okay, so water will leave your body to help equalize the concentration gradients. So when water leaves your body, the solute concentration in your body goes up, and the solute concentration in the pool goes down. Okay, and that's why you get pruning. Same thing would happen to a cell. If you put a cell in a hypertonic solution, a solution where there's a lot of solute dissolved in the solution, then water would leave the cell and the cell would shrivel and could die. A hypotonic solution would be, say you jumped in a pool that was just 100% pure water. Just pure water, not a single ion or molecule dissolved in it. That there's less solute in the pool than in your body, so water will flow into your body and you would swell. You would become swollen. That would happen to a cell as well. So if you dropped a cell in a hypotonic solution, it would swell and maybe pop. Okay? Now, when the concentrations of solute are the same on either side of a membrane, you have an isotonic solution. And the net flow of water into and out of the cell is equal, so the cell is at homeostasis. And that's one of the functions of the body, to maintain homeostasis, to maintain the isotonicity of the interstitial fluid. To keep the interstitial fluid, the fluid in between all the cells, isotonic. So cells aren't shriveling or popping, expanding, swelling. There's a good picture of that figure 3.9. Okay, so there's 4 types of diffusion. The first is simple diffusion, and before I get into that, I want to read a bit from the book here about diffusion. So diffusion is the tendency of molecules or ions to move from an area where they are in higher concentration to an area where they are in a lower concentration. That is down or along their concentration gradient. So that's an important concept right there to figure out. Molecules move down, molecules or ions move down their concentration gradient. They move from areas of high concentration to areas of low concentration. We talked about that with the salt tablet and water. The constant random and high-speed motion of molecules and ions results in collisions. With each collision, the particles ricochet off one another and change direction. The overall effect of this erratic movement is to scatter or disperse the particles throughout the environment, so basically to diffuse. The greater the difference in concentration gradient of the diffusing molecules and ions between the two areas, the more collisions occur and the faster the net diffusion of particles. So another, just kind of not really important for testing purposes, but anything that increases or affects this kinetic energy of these molecules, of these water molecules moving around and zinging around, will increase diffusion. So diffusion is faster when you add heat, right? Because kinetic energy, that is what heat is molecules moving more. So that would explain why it's easier to dissolve something in boiling water than cold water. Okay? And also why you would, okay, simple diffusion. I'm going to read just this paragraph here. In simple diffusion, nonpolar and lipid-soluble substances diffuse directly through the lipid bilayer. So we've talked about that. We've talked about how steroids and lipid molecules will flow right into a cell because momentary openings in the polar heads allow these fatty substances to slip through the plasma membrane. And of course, the lipid interior won't stop them because they're lipids, right? It just flows right through. And then those openings on either side of the polar component of the membrane allow things to slip through. But large polar molecules can't get through the membrane, so they can't participate in this. Such substances include oxygen, carbon dioxide, and fat-soluble vitamins, because they're hydrophilic, fat-soluble vitamins. Because oxygen concentration is always higher in blood than in tissue cells, oxygen continuously diffuses from the blood into the cells. Makes sense, right? Carbon dioxide, on the other hand, is in higher concentration within the cells, so it diffuses from tissue cells into the blood. These things are able to passively move through the membrane, so they're just going to move along their concentration gradients. If the concentration of oxygen is higher on the outside, it will diffuse down its gradient to the inside of the cell. Okay. Let's talk about facilitated diffusion. I'm just going to read this out of the book. Certain molecules, notably glucose and other sugars, some amino acids and ions, are transported passively even though they are unable to pass through the lipid bilayer. So even though their concentration gradients won't just force them in, sorry, let me rephrase that. Their concentration gradients are telling them to come in, but they can't get through. They can't get through the membrane because they're polar. So their concentration gradients are trying to push them into the cell, but they can't get in. All right? Instead, they move through the membrane by a passive transport process called facilitated diffusion, in which the transport substance either 1 binds to protein carriers in the membrane and is ferried across, or 2 moves through water-filled protein channels. Okay, so the driving force here is still a concentration gradient, so no energy is needed. We don't need ATP. If there's a high concentration of a certain amino acid or sugar on the outside of the cell, it's being driven into the cell. It just can't get through the membrane, so it has to go through a channel, a protein channel, or a protein carrier. Okay? Carrier-mediated facilitated diffusion. So this is the second of the two things that I was talking about. Carriers are transmembrane integral proteins that are specific for transporting certain molecules or classes of molecules that are too large to pass through the membrane channels. Okay? And I'm not going to read any farther. I think you guys can read that if you don't understand that. There's two types of facilitated diffusion. One where it goes through a channel, and that's channel-mediated facilitated diffusion. And one where it goes through a carrier, and that's carrier-mediated facilitated diffusion. And that is it for the passive processes. Let's talk about, okay, active transport. So, there's a few different types, but they all have one thing in common with facilitated diffusion, and that is that they utilize proteins. Sorry, except for vesicular transport. Primary and secondary active transport utilize integral proteins as pumps to pump substances from one side of the membrane to the other. The big difference here is that they're pumping substances against their concentration gradient. Because of that, they need an energy source besides kinetic energy, which is the case with the concentration gradient, right? Molecules are being shoved into the cell in passive transport, so it has kinetic energy as an energy source to drive that. With active transport, they're going to utilize the energy stored in the bonds between the phosphate molecules between the phosphate molecules of the ATP phosphate chains. You don't even know that. Sorry, I made that more confusing than it really is. But when we talk about primary active transport, a lot of times we're going to use the sodium-potassium pump as an example. And I'm just going to read this directly from the primary active transport section, and then I'll try to clarify a little bit. Okay. In primary active transport, hydrolysis of ATP results in the phosphorylation of the transport protein. All that really means is that an ATP binds to the protein, so it's phosphorylated. This step causes the protein to change its shape in such a manner that it pumps the bound solute across the membrane. Primary active transport systems include calcium and hydrogen pumps, especially in the stomach. You have hydrogen pumps to maintain acidity. But the most investigated example of a primary active transport system is the sodium-potassium pump, for which the carrier or pump is an enzyme called sodium-potassium ATPase. Again, I'm not going to ask you that. That's just the name. That's just the name of the pump. In the body, this is very important though. In the body, the concentration of potassium inside the cell is 10 times higher than that outside. Okay, so what does that mean? That means that potassium wants to leave the cell. Okay, and the reverse is true for sodium. Okay, so sodium is 10 times higher outside of the cell than inside, so sodium wants to enter the cell. These tonic concentration differences are essential for excitable cells like muscle and nerve cells to function normally, and for all cells to maintain their normal fluid volume. Because sodium and potassium leak slowly, but continuously through leakage channels in the plasma membrane along their concentration gradients. Okay, so what does that mean? That means that there's ways for potassium to get out and sodium to get in. So they're going to, right? Because potassium is trying to leave and sodium is trying to enter. So slowly over time, if we didn't do anything about that, these concentration gradients would equalize, okay? There'd be the same amount of potassium concentrations inside and outside, same with sodium. That's not good. That'd be bad news. Then our nerves and muscles wouldn't be able to function, including our heart. Fluid volumes would change, so that's bad. So we need an active process that will maintain this concentration gradient, okay? All right, where was I? Okay. The sodium-potassium pump operates almost continuously as an antiporter, okay? An antiporter. We'll talk about that in a second. Sorry, I lost my spot. Okay, it simultaneously drives sodium out of the cell against a steep concentration gradient and pumps potassium back in. So it's pumping against a 10-fold difference in concentration. Earlier we said that solutes diffuse down their concentration gradients. This is true for uncharged solutes, but only partially true for ions. Why? Okay, ions have one more component, right? The negatively and positively charged faces of the plasma membrane can help or hinder diffusion of ions driven by a concentration gradient, right? Because like charges repel and opposite charges attract. It is more correct to say that ions diffuse according to electrochemical gradients, thereby recognizing the effect of both electrical and concentration forces. Hence, the electrochemical gradients maintained by the sodium-potassium pump underlie most secondary active transport of nutrients and ions that are crucial for cardiac, skeletal, and neuron function. Basically, this is saying that when it comes to ions are structures that have charge. When it comes to ions, it's not just concentration that's dictating what direction they flow across the membrane. It's also electrical charge. Okay. So, I think that's it for primary active transport. We're going to talk about it more in lecture on Monday, but for now that's good. Let's talk about secondary. All right, again, I'm just going to read from the book. I hope this format's all right for you guys. I think I'm just going to keep sort of reading sections and explaining them out of the book along with the PowerPoint. All right, secondary active transport. A single ATP-powered pump, such as the sodium and potassium pump, can indirectly drive the secondary active transport of other solutes. By moving sodium across the membrane against this gradient, the pump stores energy. Okay? So does that make sense? By maintaining this strong concentration gradient, forcing sodium into the cell, right? Because it's 10 times the amount of sodium outside the cell as inside. And the sodium-potassium pump maintains that. Because the sodium leaks into the cell, it pumps it back out. Okay? By doing that, it's storing potential energy, right? Because the sodium is trying to get back in. So it stores energy in an ion concentration gradient. Then, just as water pumped uphill can do work as it flows back down, a substance pumped across the membrane can do work as it leaks back in. Because remember, there's leakage channels. So as the sodium's pumped out, it's flowing back in at the same rate. Okay? So the sodium pump's maintaining the gradient, but that sodium's still flowing back in. It gets pumped back out, but it flows back in. A substance pumped across the membrane can do work as it leaks back, whereas it's propelled downhill along its gradient. In this way, as sodium moves back into the cell with the help of a carrier protein, other substances are dragged along or co-transported by the same carrier protein. This is a Symport system. For example, some sugars, amino acids, and many ions are co-transported via secondary active transport to cells lining the small intestine. Because the energy for this type of transport is a concentration gradient of the ion, in this case sodium, sodium has to be pumped back out of the cell to maintain its diffusion gradient. Ion gradients can also drive antiport systems, such as those that help regulate intracellular pH by using the sodium gradient to expel hydrogen ions. An antiport system is when let's look at this example. As sodium comes in, is forced into the cell, that pump, after the sodium comes in, that pump reverses and expels the hydrogen ion. Okay, so that's an antiport system. It's the system where instead of bringing substances along with the carrier ions, in this case sodium, along with it, down its concentration gradient, it's propelling things against. So as it comes in, it's expelling things out. Okay. Regardless of whether the energy is provided directly, primary active transport, or indirectly, secondary active transport, each membrane pump or co-transporter transports only specific substances. Active transport systems provide a way for the cell to be very selective in cases where substances cannot pass by diffusion. No pump, no transport. All right. So let's get into vesicular transport. A vesicle is a little sphere made up of plasma membrane. Okay, it's a little piece of the plasma membrane folds in, invaginates, and then pinches off and makes a sphere that can contain something. Let's get into. Let's just read it. Okay. In vesicular transport, fluids containing large particles and macromolecules are transported across cell membranes inside membranous sacs called vesicles. Like active transports, vesicles transport substances. Like active transport, vesicular transport moves substances into the cell and out of the cell. Okay, so when it's bringing substances into the cell, it's called endocytosis in the cell. When it's expelling substances, that's called exocytosis. It is also used for combination processes such as transcytosis, which basically means it brings something in, moves it to the other side of the cell, and expels it. So this might be in the intestine when you're bringing substances in, and then you're pumping them out the other side of the cell. All these processes are energized by ATP, or in some cases GTP, which is guanosine triphosphate. We're not going to get into that very much. All right, so let's look at this first bullet here: endocytosis, transcytosis, vesicular trafficking. The figures in your guys' book are really good. I've tried to include some on here, so I've definitely tried to check those out or look at them as you're listening to this. Virtually all forms of vesicular transport involve an assortment of protein-coated vesicles of 3 types, and with a few exceptions are all mediated by membrane receptors. That makes sense. You need a receptor so the cell knows whether or not to start this process. Okay? When a certain substance binds to a receptor, that triggers something. If you're in a cell that, say, in your small intestine that's bringing substances in, and sugar comes by and binds to its receptor, it will bring it in. All right, so general scheme of endocytosis. Protein-coated vesicles provide the main route for endocytosis and transcytosis of bulk solids, most macromolecules and fluids. On occasion, these vesicles are also hijacked by pathogens seeking entry into the cell. So some pathogens have figured out a way to look like the target chemical, and then they bind to that receptor, and then they get taken in, and then they hijack the cell. All right. Step 1 of endocytosis. An infolding portion of the plasma membrane called a coated pit progressively encloses a substance to be taken into the cell. I'm skipping some stuff. All right, that's okay. So step 1, the plasma membrane starts to fold and create a coated pit, which is a pit coated in proteins and receptors. And the protein is clathrin. The clathrin coat and some accessory proteins act in both selecting the cargo and deforming the membrane to produce a vessel. So they select the cargo, and they're actually what causes the membrane to enfold and capture the substance. Step 2, the vesicle detaches And so that's step 2. The vesicle detaches. And step 3, the co-proteins are recycled back into the membrane. So those proteins are recycled. A lot of things in the cell are recycled. To be more efficient. The uncoated vesicle then typically fuses with a sorting vesicle called an endosome. So this is another vesicle. And they bind together to make a bigger vesicle, and then they're sorted. Some membrane protein components and receptors of the fused vesicle may be recycled back to the membrane in a transport vesicle. Step 6, the remaining contents of the vesicle may either combine with the lysosome, which is a digestion vesicle, which we'll talk about later, and that will degrade the substance and then release it. This is usually for iron or cholesterol. Or B, it may be transported completely across the cell and released by exocytosis on the opposite side. And we talked about that a little bit. That's transcytosis. So that's when you need to move chemicals from one side of the cell to the other. Here we go. This is common in the endothelial cells lining blood vessels because it provides a quick means to get substances from the blood to the interstitial fluid. Okay, some types of endocytosis are phagocytosis. So that's when, white blood cells and phagocytes, macrophages, they can destroy invaders. And phagos basically means eat. They can eat debris, other cells, bacteria, dead cells, things like that. Penocytosis is kind of the equivalent of drinking something in the world of cells. They're bringing fluid only, or mostly fluid. And then receptor-mediated endocytosis, which is what we've been talking about, and that's where certain substances bind to receptors and then they're taken into the cell. And I'm not going to go into the 3 types much other than that. All right, so exocytosis. And guys, these figures are good. Figure 312 on page 76, check that out. Look at the 6 steps. Those 6 steps will be important. And same thing with 313. I think if you look at the right side of figure 13 is pretty cool, how the snare proteins bind and things. I'm not going to go into it here, but check that out. It's cool. All right, exocytosis. Vesicular transport processes. Vesicular transport processes that eject substances from the cell interior into the extracellular fluid are called exocytosis. All right. So basically, endocytosis in reverse. All right. This is ejecting things into the interstitial fluid. Typically stimulated by a cell surface signal such as binding of a hormone to a membrane receptor or a change in the membrane voltage. Exocytosis accounts for hormone secretion, neurotransmitter release, mucus secretion, and in some cases ejection of waste. The substances to be removed from the cell is enclosed in a protein-coated membranous sac, which is now called a secretory vesicle. In most cases, the vesicle migrates through the plasma membrane, fuses with it, ruptures, and spills its contents into the cell. That's good. Let's go to this next thing, which is resting membrane potential. This is really important to understand, not just for this next week's quiz and then the test, but for other chapters we're going to be talking about, especially next quarter, when we talk about heart and nerve cells. In this quarter, when we talk about muscle cells. All right, guys. Here goes. So earlier when we were talking about, I'm going to read from the book in a second, but earlier when we were talking about how a active transport pump, like the sodium-potassium pump, is creating a potential. It's storing potential energy, right? It's creating a concentration gradient and therefore storing potential. We call that a membrane potential. Specifically with ions, because we're creating not only a concentration gradient, but an electric gradient, or put together, like we talked about earlier, an electrochemical gradient. So let's just go into this paragraph here and then talk about it a little bit after. As you're now aware, the selective permeability of the plasma membrane can lead to dramatic osmotic flows, but that is not its only consequence. An equally important role is generation of a membrane potential, or voltage, across the membrane. A voltage is electrical potential energy resulting from the separation of oppositely charged particles. In cells, the oppositely charged particles are ions, and the barrier that keeps them apart is the plasma membrane. So I don't know about you guys, but electricity is something that's always been a little elusive for me to understand. Current, voltage, ohms, resistance, these different types of things. But in this case, and this has helped me understand what voltage is the potential for electricity to flow. Okay? So when we have an electrical potential, there's a potential for, and this is electrochemical. So let me back up. Let's say we look at sodium. It's an ion. It has a positive charge. That means that because there's such a high concentration of sodium outside of the cell, its concentration gradient is forcing it in the cell, but it's also being forced in the cell by an electrical gradient because the positive ions are repelling each other. Okay, so that potential can be measured in voltage. How strongly is that sodium being pushed into the cell by its electrical gradient? Okay. And the reason this potential exists is because the plasma membrane isn't letting them cross. Okay, the sodium-potassium pump is maintaining the concentrations. The potential at rest is relatively fixed at a certain level, and you can measure that in volts. Okay. In the resting state, all-body plasma membranes exhibit a resting membrane potential that typically ranges from -50 to -100 millivolts, depending on cell type. For this reason, all cells are said to be polarized. The minus sign before voltage indicates that the inside of the cell is negative compared to its outside. Alright, so the inside of the cell membrane is negative compared to the outside. And the difference in that charge is measured in a voltage. In this case, the difference is between, at rest in those cells, between -50 to -100. So there's a difference of 50 to 100 millivolts on either side of the cell. That's the voltage. Okay, the positive environment on the outside wants to move inwards towards the negative environment, and the negative environment on the inside wants to move outwards towards the positive, but it's not because the cell membrane is separating them. So that is a electrical potential. Super important. And when we look at a cell at rest, we're looking at its resting membrane potential, which is between -50 and -100 millivolts. Alright, this voltage or charge separation exists only at the membrane. Okay? What does that mean? It means that the average of all the ion charges inside the cell and outside the cell are actually the same. The difference is only at the membrane. Okay, let me read that. If we added up all the negative and positive charges in the cytoplasm, we would find that the cell interior is electrically neutral. Likewise, the positive and negative charges in the extracellular fluid balance each other out exactly. So how does the resting membrane potential come about, and how is it maintained? The short answer is that diffusion causes ionic imbalances that polarize the membrane, and active transport processes to maintain that membrane potential. First, let's look at how diffusion polarizes the membrane. Okay, diffusion, before I get into the paragraph again, diffusion is concentration gradients. Okay? Those concentration gradients create a electric potential, and an active transport process is maintained. Many kinds of ions are found both inside cells and in the extracellular fluid, but the resting membrane potential is determined mainly by the concentration gradient of potassium, which is a positive ion, like sodium, and by the differential permeability of the plasma membrane to potassium and other ions. And figure 3.15 is really good to look at for this. Actually, the figure in the PowerPoint is pretty similar, so you can check that out. Recall that potassium and protein anions predominate inside body cells. Alright, so there's a high concentration of potassium, which is positive, and a high concentration of proteins with negative charges inside the cell. Okay, and they're attracted to each other. The potassium and the negative anions, protein anions, are attracted to each other. The extracellular fluid contains relatively more sodium, which is largely balanced by chloride. So basically dissolved salt on the outside of the cell. The unstimulated plasma membrane is somewhat permeable to potassium because of leakage channels, but impermeable to the protein anions. So potassium can leave, but the negative anions can't. So the concentration gradient is forcing potassium out, but the protein anions are pulling it back. Consequently, potassium diffuses out of the cell along its concentration gradient, but the protein anions are unable to follow. And this loss of protein charges makes the membrane interior more negative. So the concentration gradient for potassium is stronger than its electrical gradient that's holding it back to the anions. So potassium leaves, but the anions remain right at the plasma membrane interior. So the plasma membrane interior is negative because of all the negative proteins. Alright, there's a little step list here. So step 1, as more and more potassium leaves the cell, the negativity of the inner membrane face becomes great enough to attract potassium back towards and even into the cell. At a membrane voltage of -90 millivolts, potassium's concentration gradient is exactly balanced by the electrical gradient, and a resting membrane potential is achieved. And one potassium enters the cell as one leaves, so there's a net flow of potassium. So the charge separation doesn't become strong enough to hold back the concentration gradient of potassium until -90 volts. Okay, so enough potassium has to leave, enough positive potassium has to leave to make the cell's interior right along the plasma membrane have a -90 millivolt difference in electricity, in the electrical gradient. Hope that makes sense. If that doesn't make sense after the lecture on Monday, or I guess you'll be listening to this after the lecture. If it doesn't make sense after lecture and this, try to make sure to get a hold of me, okay? Alright, step 2, I just wrote step 2. Step 3, in many cells, step 3 was at this net balance, so that's it. That's on the top left of page 80 if you want to read through that again. In many cells, sodium also contributes to the resting membrane potential. Sodium is strongly attracted to the cell's interior by its concentration gradient, bringing the resting membrane potential to -7. However, potassium still largely determines resting membrane potential because the membrane is much more permeable to potassium than sodium. Even though the membrane is permeable to chloride, in most cases chloride does not contribute to the resting potential because its concentration and electrical gradients exactly balance each other. You may be tempted to believe that massive flows of potassium ions are needed to generate the resting potential, but this is not the case. Surprisingly, the number of ions producing the membrane potential is so small that it does not change ion concentrations in any significant way. So even though all this flow of potassium is happening at the membrane, overall, the change of potassium is almost non-existent, okay? These are just a relatively small amount of ions occurring just right at the membrane surface on the interior and exterior. And it doesn't seem that important, but when it comes to transmitting electrical signals along nerves and muscles in the heart, it's very important. And differences in these ion concentrations can throw that off. In a cell at rest, very few ions cross its membrane. However, sodium and potassium are not in equilibrium, and there is some net movement of potassium out of the cell and sodium into the cell. Sodium is strongly pulled into the cell by both its concentration gradient and its electrical gradient. If only passive forces were at work, these ion concentrations would eventually become equal inside and outside of the cells. But they don't because of active transport, sodium, potassium pump, and other types of pumps. Oh, and that's the next section. Also, active transport maintains electrochemical gradients. Now let's look, this is out of the book. Now let's look at how active transport processes maintain the membrane potential that diffusion has established. With the result that the cell exhibits a steady state. The rate of active transport is equal to and depends on the rate of sodium diffusion into the cell. If more sodium enters, more is pumped out. This is like being in a leaky boat. The more water that comes in, the faster you bail. The faster you get rid of the water. The sodium-potassium pump couples sodium-potassium transport, and on average, each turn of the pump ejects 3 sodium out of the cell and carries 2 potassium back in. Because the membrane is always 50 to 100 times more permeable to potassium, the ATP-dependent sodium-potassium pump maintains both the membrane potential, which is the charge separation, and the osmotic balance, the concentration gradient. Indeed, if sodium was not continuously removed from the cells, so much would accumulate intracellularly that the osmotic gradient would draw water into the cells, causing them to burst. water follows sodium, right? So as sodium moves into the cell, so would water, and the cells would burst. So active transport prevents that. This gets into why high sodium diets cause high blood pressure and things like that too, but we won't. That's for a later date. As we described on page 73, diffusion of charged particles across the membranes is affected not only by concentration gradients, but by the electrical charge in the inner and outer faces of the membrane. Together, these gradients make up the electrochemical gradient. The diffusion of potassium across the plasma membrane is aided by the membrane's greater permeability to it and by the ion's concentration gradient. But the negative charges on the cell's interior resist potassium diffusion. In contrast, a steep electrochemical gradient draws sodium into the cell, but the membrane's relative impermeability to it limits sodium diffusion. This is just describing the gradients at rest. I think we pretty much covered what's important out of that already. The transient opening of gated sodium-potassium channels in the plasma membrane upsets the resting membrane potential. Okay, and this is talking about something we're not going to be talking about for a few chapters, but we can change this resting membrane. It's a resting membrane potential. We can change it. And it's important that we're able to change it when we want to transmit chemical signals or nerve impulses. Okay? And that's why this whole section is important. But that's it for now. This is just resting membrane potential. Cell interactions. So we're still on the plasma membrane. We're still talking about all the things the plasma membrane does. So it does a lot more than just being structural. But I think we're just about done. A few more slides on plasma membrane. Okay, roles of plasma membrane receptors. I'm not sure if this is something I skipped on purpose. Let me read a little bit here. A huge and diverse group of integral proteins and glycoproteins that serve as binding sites are collectively known as membrane receptors. Some function in signaling and some function in contact signaling and others in chemical signaling. I'm trying to find cell adhesion molecules in the book. Okay, here we go. I kind of skipped a section. Okay. Thousands of cell adhesion molecules are found on almost every cell in the body. CAMs, cell adhesion molecules, play key roles in embryonic development and wound repair, situations where cell mobility is important, and in immunity. These sticky glycoproteins act as the molecular velcro that cells use to anchor themselves to molecules in the extracellular space and to each other. They act as the arms that migrating cells use to haul themselves past one another. SOS signals sticking out of the blood vessel lining that rally protected blood cells to a nearby infected or injured area. Mechanical sensors that respond to changes in local tension or fluid movement at the cell surface by stimulating synthesis or degradation of adhesive tight junctions. Transmitters of intracellular signals that direct cell migration, proliferation, and specialization. I don't know if we're really going to get deep into this. There's entire classes on cellular communication and things like that. I think it's sufficient if you just understand on this section that CAMs just serve to act as a way of communication between cells. All right, let's get into the role of the receptors a bit more. There's two types, and I read this on accident earlier: contact signaling and chemical signaling. All right, let's go into contact signaling really quick. This is when cells come together and touch. It is the means by which cells recognize one another. It is particularly important for normal development and immunity. Some bacteria and other infectious agents use contact signaling to identify their preferred target tissues. Okay. Chemical signaling. Most plasma membrane receptors are involved in chemical signaling. Ligands are chemicals that bind specifically to plasma membrane receptors. Ligands include most neurotransmitters, nervous system signals, hormones, and paracrines. Different cells respond in different ways to the same ligand. Acetylcholine, for instance, stimulates skeletal muscle tissues to contract, but inhibits heart muscle. Why do different cells respond so differently? The reason is that a target cell's response depends on the internal machinery that the receptor is linked to, not the specific ligand that binds to it. Though cell responses to receptor binding vary widely, there is a fundamental similarity. When a ligand binds to a membrane receptor, the receptor structure changes, and the cell proteins are altered in some way. For example, catalytic receptor proteins are membrane proteins that respond to ligands by becoming activated enzymes. Chemically gated channel-linked receptors, which are common in muscle and nerve cells, respond by transiently opening or closing ion gates, which in turn changes the excitability of the cell. So this goes back to membrane potential. Certain chemical ligands can bind to receptors and change the membrane potential. And this is how nerve impulses and muscles contract. Nerve impulses are formed and muscles contract. And then there are G protein-linked receptors. This comes up a lot in like a molecular biology class. We're just going to really briefly describe it. And that's what this image is here. G protein-linked receptors exert their effect indirectly through G protein, a regulatory molecule that acts as a middleman or a relay to activate or inactivate a membrane-bound enzyme or ion channel. Okay? So in the picture, we have a G-linked protein receptor. A ligand binds to it. The GDP is phosphorylated, a phosphate is added to GDP, it becomes a GTP molecule, or active G protein. And then it moves somewhere else and activates the protein and makes a activated enzyme. That's on step 2 there, and creates some type of cellular response. Then the phosphate leaves and we start over. All right. I think that's enough. I'm not going to keep reading through here. We'll talk about cyclic AMP and ionic calcium in the lecture. Okay. Whew. Sorry, I'm getting a little. Hey guys, so next we're going to be talking about cytoplasm. So we talked about the plasma membrane. It contains a cell and is selectively permeable double lipid bilayer that either regulates the concentration of ions and molecules passively or actively. Next, we're going to be talking about the cytoplasm. So there's 3 general components. There's the cytosol, which is the fluid. The book defines it as the viscous, semi-transparent fluid in which the other cytoplasmic elements are suspended. It is a complex mixture with properties of both a colloid and a true solution. So it's very viscous. Dissolved in the cytosol, which is largely water, are proteins, salt, sugars, and other solutes. Alright, so the solute is the fluid of the cytoplasm. And I forgot to mention, the cytoplasm is everything inside the cell membrane except for the nucleus. Okay, the second component of the cytoplasm are organelles, and these are all of the little metabolic machinery in the cell. So if we look at this picture of the composite or generalized cell, we see labeled, we see mitochondria, those are the little kidney bean things. We see green bulbous thing, that's Golgi apparatus. We have endoplasmic reticulum. All the little structures inside of the cell are the organelles. And lastly, we have inclusions. And these are just stored pockets of some type of substance. If we're in liver cells, it might be glycogen. If we're in fat cells, it might be adipose droplets. Not all cells have inclusions, but quite a few do, and it's just another component of the cytoplasm if present. Alright, so the organelles we're going to talk about, the first organelle we're going to talk about is the mitochondrion. The mitochondria of the cell are pretty interesting. I think they're probably the most interesting organelle because their origin is sort of not truly understood. And there's some really unique things about the mitochondria that we'll get to a little bit later. Let me start by reading a little bit from the book. Okay, the mitochondria is a membranous organelle, so it has a membrane just like the cell. It's a lozenge-shaped membranous organelle. In living cells, they squirm, elongate, and change shape almost continuously. They are the power plants of a cell, providing most of its ATP supply. So I remember when I learned about the mitochondria, they're always described as the power plant, the power source of the cell. I think that's fair to say, but when I think of the power plant or the powerhouse, I think of like the motor for a car, right? I think of it as the thing that actually drives movement. But in the case of the cell, that's the mitochondria don't actually make the cell move. They produce fuel for the cell's reactions. So I think of the mitochondria more of like an oil refinery or a gas station, producing the fuel that the cell needs for metabolism. Okay. The density of mitochondria in a particular cell reflects that cell's energy requirements. Okay, what does that mean? So that means cells that require a lot of energy, like muscle cells, are going to have a lot of mitochondria. Structure equals function. Mitochondria generally cluster where the action is. Busy cells like kidney and liver cells have hundreds of mitochondria, whereas relatively inactive cells, such as unchallenged lymphocytes, just have a few. And if you guys already know a bit about anatomy, you might be mad at my last analogy. Some muscle cells have a lot of mitochondria, the aerobic type 1 muscle cells, and others have a little bit, so I just wanted to clarify that. But cells like kidney and liver cells have hundreds of mitochondria because they're highly active. Okay. A mitochondrion is enclosed by two membranes, not just one, each with the general structure of the plasma membrane, and you can see this in figure 3.17. The outer membrane is smooth and featureless, but the inner membrane folds inward, forming shelf-like cristae that protrude into the matrix. So the inner membrane has lots of folds and pockets and wrinkles in it. The outer membrane is just a smooth, lozenge-shaped membrane. The cristae protrude into the matrix, a gel-like substance within the mitochondria. So even the mitochondria almost has a cytosol. They don't call it that, but they just call it gel-like substance. Intermediate products of food, fuels, glucose, and others are broken down to water and carbon dioxide by means of enzymes. Some dissolve in the mitochondrial matrix, and others form part of the cristomembrane. All right, that's the inner membrane. As the metabolites are broken down and oxidized, some of the energy released is captured and used to attach phosphate groups to ADP molecules to form ATP. This multi-step mitochondrial process is called aerobic cellular respiration because it requires oxygen. If you've taken other classes, you might have heard of this process being called the Krebs cycle, or there's a way to metabolize pyruvate that's in the cytosol, but the Krebs cycle occurs in the mitochondria. And this is basically where you add a phosphate group to an ADP or an adenosine triphosphate and make it an adenosine triphosphate. So you're adding a phosphate. Then that ATP molecule can go be used as fuel somewhere else in the cell, where that phosphate will be broken off and energy will be released. Mitochondria, I'm reading from the book again. Mitochondria are complex organelles. They contain their own DNA, RNA, and ribosomes. Okay, so that's what really makes mitochondria unique, and this is important. They're the only thing that has its own set of DNA, RNA, and ribosomes. So it's almost like it's an entirely different living entity. It's not, but it's very strange compared to the other organelles, and they're going to get into why they think this may be in the next paragraph. Okay, DNA, RNA, and ribosomes. So they contain their own DNA, RNA, and ribosomes, and are able to reproduce themselves. Mitochondrial genes, there's about 37 of them, direct the synthesis of 1% of the proteins required for mitochondrial function. And the DNA of the cell's nucleus encodes the remaining proteins needed to carry out cellular respiration. When cellular requirements for ATP increase, the mitochondria synthesize more cristae or simply pinch in half, a process called fission, to increase their number and then grow to their former size. Intriguingly, mitochondria are similar to bacteria in the purple bacteriophylum. So if you look at them, they kind of look like what we've sort of been taught that bacteria look like. They're kind of a kidney bean lozenge shape. And that's what the book's saying. Interestingly, mitochondria are similar to bacteria. Sorry guys, I can't talk right now. And mitochondrial DNA is a lot like bacterial DNA. It is widely believed that mitochondria arose from bacteria that invaded the ancient ancestors of plant and animal cells, and that this unique merger gave rise to all complex cells. So wow, that's really interesting, right? So basically what they're saying is that in early cell life, whatever you believe that was, mitochondria were bacteria that invaded cells, and then they sort of created this symbiotic relationship where the mitochondria lived within the cells and reproduced, and generation after generation stayed in these cells and gave rise to our complex cells. So I think that's pretty crazy, but it's definitely the only organelle that has those features, and that might be important for a test coming up because it's very unique. Okay, next organelle. We're going to be talking about ribosomes. If you've taken any science or biology, at least classes in the past, you probably learned that ribosomes are the site of protein synthesis. Okay. Let's get into the structure of ribosomes a bit. We're going to talk about protein synthesis in some pretty great detail later from the book. Ribosomes are small, dark-staining granules composed of proteins and a variety of RNAs called ribosomal RNAs. Each ribosome has two globular subunits that fit together like the body and cap of an acorn. Ribosomes are sites of protein synthesis, a function we discuss in detail later in this chapter. I guess I didn't need to say that earlier. Some ribosomes float freely in the cytoplasm. Others are attached to membranes, forming a complex called the rough endoplasmic reticulum. These two ribosomal populations appear to divide the core of protein synthesis, the chore of protein synthesis. Free ribosomes float freely in the cytoplasm. They make soluble proteins that function in the cytosol, as well as those imported into the mitochondria and some other organelles. Membrane-bound ribosomes are attached to membranes, forming a complex called the rough endoplasmic reticulum. They synthesize proteins destined either for incorporation into cell membranes or lysosomes, or for export from the cell. Ribosome subunits can switch back and forth between these two functions, attaching to and detaching from the membranes of the endoplasmic reticulum according to the type of protein they're making at a given time. So basically, there's ribosomes that float around in the cytosol, and there's ribosomes that are bound to the rough endoplasmic reticulum. They both make proteins that are destined for different sites of the cell. I'll ask you which proteins make which types of proteins. Which ribosomes make which type of proteins. I just want you to know that they exist. They're made of RNA and amino acids, and they can be found either at the rough ER, endoplasmic reticulum, or in the cytosol. All right, so let's segue into the rough. Oops, I got a little ahead of myself. Let's segue into the endoplasmic reticulum. There's two types. There's a smooth endoplasmic reticulum and the rough endoplasmic reticulum. Really, the difference between these is that one has ribosomes attached to it and one doesn't. So one's rough, the one with ribosomes, one's smooth. There's actually more differences than that. Because ribosomes are the site of protein synthesis, it makes sense that proteins are produced and stored and packaged and transported within the rough ER. In the smooth ER, there's no ribosomes, so that means there's no protein. So they're responsible for lipid metabolism, hormone metabolism, and lipid storage and packaging, processing, things like that. They're also a little bit different in shape. The smooth endoplasmic reticulum is very tubular, and the rough has flattened sacs and is more folded. You're going to see a lot of smooth ER in cells like muscle cells because the smooth endoplasmic reticulum can also break down stored glycogen to form free glucose. So cells that use a lot of glucose will have a lot of smooth endoplasmic reticulum. All right. Let's get into smooth ER. So the smooth ER is continuous with the rough ER and consists of tubules arranged in a looping network. Its enzymes play no role in protein synthesis. Okay, that's important. Instead, the enzymes catalyze reactions involved with the following tasks: metabolizing lipids, synthesizing cholesterol, and synthesizing the lipid components of lipoproteins. That occurs in liver cells. again, I won't on the testing quizzes. I won't get into that level of detail. I want you guys to understand these things conceptually. It also synthesizes steroid-based hormones such as sex hormones. It absorbs, synthesizes, and transports fats. This happens in intestinal cells especially. Detoxifies drugs, certain pesticides, and cancer-causing chemicals. Those happen in your organs that are responsible for detoxifying blood, and those are the liver and kidneys. And then also breaks down stored glycogen to form free glucose, in liver cells especially. I said muscles earlier. I said that a little bit backwards. The smooth endoplasmic reticulum breaks down glycogen to form glucose in the liver, which is then absorbed by the muscle cells. The glucose is then absorbed by muscle cells. All right, that's it for smooth ER. We'll talk about smooth ER a lot more when we get to the muscles and other sections. Right now, we'll probably spend a bit more time in the rough ER. So, the rough endoplasmic reticulum. The external surface of the rough ER is studded with ribosomes, hence the name rough. Proteins assembled on these ribosomes thread their way into the fluid-filled interior of the ER cisterns. So, unlike the cytosol ribosomes, which just kind of spit proteins out into the cytosol, when proteins are transcribed here, when they're synthesized here, those amino acid chains, those proteins enter the ER to be sorted and packaged. I might have already read this, but proteins assembled on these ribosomes spread their way into the fluid-filled interior of the ER cisterns. These are these hollow caverns. When complete, the newly made proteins are enclosed in vesicles for their journey to the Golgi apparatus, where they undergo further processing. The rough ER has several functions. Its ribosomes manufacture all proteins secreted from cells. For this reason, the rough ER is particularly abundant and well developed in most secretory cells, antibody-producing plasma cells, and liver cells, which produce most blood proteins. It is also the cell's membrane factory, where integral proteins and phospholipids that form part of the cellular membranes are manufactured. Okay, so why is that necessary? So we talked about exocytosis and all these other functions which basically require portions of membrane. So if you're going to get rid of something, if you're going to excrete something, and you surround that substance by the cell's plasma membrane, and then it pinches off and is expelled, the cell just got smaller because you just got rid of plasma membrane. Well, plasma membrane is produced in the rough endoplasmic reticulum. It then pinches off and makes a vesicle and moves out and forms with the cell's membrane, making the cell bigger. It's also important in cellular growth. When the cell needs to get bigger, it needs a source of membrane to get bigger. And that comes from the rough endoplasmic reticulum. Another important thing. The enzymes needed to catalyze lipid synthesis have their active sites on the external face of the ER membrane, where the needed substrates are readily available. You can just ignore that for my class at least. Alright, so basically the rough ER has ribosomes on it. The ribosomes make proteins. The proteins are folded or modified or whatever. They're then bound in vesicles. So just like exocytosis out of the cell, but this is exocytosis out of the rough ER. So they're surrounded by membrane, it splits away, and then it's transported using motor proteins along actin chains, which we'll talk about a bit later. It's literally moved with a little motor protein over to the Golgi apparatus. The Golgi apparatus is like a giant FedEx facility. It packages, moves, and determines where these vesicles are going to go and what's going to happen to these proteins. Are they going to go to the plasma membrane to be integrated into the membrane? Maybe they're going to be sodium-potassium pumps. Maybe they'll be expelled from the cell to be used somewhere else. Or maybe I don't know. We'll get it. We'll read from the book and see. All right. So next slide. The Golgi apparatus. The Golgi apparatus consists of stacked, inflated membranous sacs, shaped like hollow dinner plates, associated with swarms of tiny membranous vesicles. So you have swarms of vesicles coming into the Golgi apparatus, and then swarms of vesicles leaving it. So the swarm is coming from the ER, and then whatever's done to the protein is done, and then a bunch vesicles leave to go to their final destination. The Golgi apparatus is the principal traffic director for cellular proteins. Its major function is to modify, concentrate, and package the proteins and lipids made at the rough endoplasmic reticulum and destined for export from the cell. The Golgi's odd shape is a side effect of the job. The protein complex pulls membranous sacs containing newly synthesized proteins off the Golgi, and in the process, the membranes are flattened like rubber bands. Transport vesicles that bud off from the rough ER move to infuse with the membranes at the convex cis-face, the receiving side of the Golgi apparatus. Inside the apparatus, the proteins are modified. Some sugar groups are trimmed while others are added. In some cases, phosphate groups are added. The various proteins are tagged for delivery to a specific address, sorted and packaged in at least 3 types of vesicles that bud from the concave trans face, the shipping side of the Golgi sac. Vesicles containing proteins destined for export pinch out from the transface as secretory vesicles or secretory granules, which migrate to the plasma membrane and discharge their contents from the cell by exocytosis. This is pathway A. Specialized secretory vesicles, such as the enzyme-producing cells of the pancreas, have a prominent Golgi apparatus. So cells that release a lot of things are going to have a well-developed rough ER and a well-developed Golgi apparatus. Okay. The Golgi apparatus pinches off other vesicles containing lipids and transmembrane proteins destined for the plasma membrane, which is pathway B, or for other membranous organelles like the mitochondria. The Golgi apparatus also packages digestive enzymes into membranous lysosomes that remain in the cell, pathway C. Lysosomes are another type of organelle, which we'll discuss in a second. But before we move on, just in summary, the Golgi apparatus, it packages, sorts, and decides where these proteins that are being made at the ER are going to go. So it's a big post office, basically. Or I guess what they say, a traffic director. So, important things on this that get brought up in quizzes and tests a lot are the cis and the transface. So, the transface is the exporting side, and that's easy to remember. Transport, things are being transported away from. The receiving side is the cis face. I'm sure in Latin that means something. I'm not sure, but I just remember that the transface is the side that's transporting away, and then it's easy for me to just process of elimination. Remember that the cis face is the receiving side. All right, zones. And guys, I think this is a format I'm going to stick with, where I read and just kind of summarize. I'm trying to get smoother as I go along. I'm thinking this will probably be the worst the worst PowerPoint webinar, and they'll just get better from here, so stick with me, and I hope you learned something from it. So 2 more types of cytoplasmic organelles are peroxisomes and lysosomes. Peroxisomes are spherical membranous sacs containing a variety of powerful enzymes, the most important of which are oxidases and I never know how to pronounce this, if it's catalases or catalysis. I think it's catalases. So they contain oxidases and catalases. Oxidases use molecular oxygen to detoxify harmful substances, including alcohol and formaldehyde. Their most important function is to neutralize free radicals, highly reactive chemicals with unpaired electrons that can scramble the structure of biological molecules. Oxidases convert free radicals to hydrogen peroxide, which is also reactive and dangerous, but which the catalases quickly convert to water. So the catalases convert H2O2 into H2O. Free radicals and hydrogen peroxide are normal byproducts of cellular metabolism, but they have devastating effects on cells if allowed to accumulate. Peroxisomes are especially numerous in liver and kidney cells. Makes sense, right? The liver is basically what detoxifies and manages the chemicals in our blood, and one of the things peroxisomes metabolize is alcohol. So it makes sense. They also play a role in energy metabolism by breaking down and synthesizing fatty acids. Peroxisomes look like small lysosomes, which we'll talk about in a second. And for many years, it was thought that they were self-replicating organelles formed when existing peroxisomes simply pinch in half. Recent evidence, however, suggests that most new peroxisomes form by budding off of the endoplasmic reticulum via a special ER machinery that differs from that used for vesicles destined for modification of the Golgi apparatus. So that was a long paragraph just saying that peroxisomes come from the endoplasmic reticulum, and they have a unique and complex pathway that allows for that to take place. Lysosomes. Born as endosomes, which contain inactive enzymes, lysosomes are spherical membranous organelles containing activated digestive enzymes. As you might guess, lysosomes are large and abundant in phagocytes. Why might that be? So we have a macrophage or a phagocyte, some type of white blood cell that's going to engulf other cells or other invaders using phagocytosis, which is just basically a really large-scale version of endocytosis. So they take in a foreign invader. After you take in a foreign invader, that invader is in a vesicle, right? Because you surround that invader by plasma membrane, and then you take that vesicle, that membranous sac, into the cell. If you just left it alone, that invader might ultimately kill the white blood cell or hijack it. So we need some type of digestive vesicle that can bind to the endosome, to the vesicle that was taken in, and digest it. Okay, so lysosomes are large and abundant in phagocytes, the cells that dispose of invading bacteria and cellular debris. Lysosomal enzymes can digest almost all kinds of biological molecules. They work best in acidic conditions and are so called acid hydrolysis. Sorry guys, I've been talking for a while. The lysosomal membrane is adapted to serve lysosomal functions in two important ways. First, it contains the hydrogen, an H plus atom, which is really just a proton. So H plus pumps, which are ATPases that gather hydrogen ions from the surrounding cytosol to maintain the organelle's acidic pH. This is just saying that they remain acidic by pumping in hydrogen ions. Hydrogen atoms, protons, basically, proton pumps. I don't know if you remember, but adding hydrogen to a fluid makes it acidic. It creates hydronium, which is H3O, so H2O with an added hydrogen, or an added proton rather, and that's very acidic. Second, it retains the dangerous acid hydrolases while permitting the final products of digestion to escape so that it can be used by the cell or excreted. So this allows digestion to occur safely. These digestive enzymes are not released into the cytosol, but rather just the digested whatever that was taken into the cell is released to be used or recycled or whatever. Basically, and this is from the book, lysosomes function as a cell's demolition crew by digesting particles taken in by endocytosis, particularly bacteria, viruses, and toxins, degrading worn-out or non-functional organelles, performing metabolic functions such as glycogen breakdown, breaking down non-useful tissues, such as the webs between the fingers and toes of a developing fetus, it's kind of interesting, and breaking down bone to release calcium ions into the blood. So in a few weeks, we'll talk about something called osteoclasts, which are cells that break down bone. There's a lot of lysosomes in these cells. Again, just another example of how an anatomy structure equals function. I guess we'll read this. The lysosomal membrane is ordinarily quite stable, but it becomes fragile when the cell is injured or deprived of oxygen and when excessive amounts of vitamin A are present. When lysosomes rupture, the cell digests itself, a process called autolysis. Autolysis is the basis for desirable destruction of cells, as in the fourth listed item above. So, in the cells in our webs, in our hands, in our fetus, the cells autolyse, and that just destroys the cells, is all that section's saying. Alright, let's skip the next couple paragraphs in your Merriam book and go over to cytoskeleton, which is another cytoplasmic organelle. Alright, the cytoskeleton, literally cell skeleton, is an elaborate network of rods running through the cytosol. and hundreds of accessory proteins that link these routes to other cell structures. It acts as the cell's bones, muscles, and ligaments by supporting cellular structures or organelles and providing the machinery to generate various cell movements. The three types of rods in the cytoskeleton are microfilaments, intermediate filaments, and microtubules. None of these are membrane covered. So these are not membranous organelles, but they are cellular organelles still, and they form a cell skeleton, a cytoskeleton, and it's a lot like our skeleton. It supports the body. It provides a place for muscles. They're not muscles in the cell, they're motor proteins, but for muscles to create movement along and across. They resist tensile forces. They give the cell structure and shape and strength. Let's start with microfilaments. These are the thinnest elements of the cytoskeleton. They're semi-flexible strands of the protein actin. Each cell has its own unique arrangement of microfilaments, so no two cells are alike. However, nearly all cells have a fairly dense cross-linked network of microfilaments called the terminal web, attached to the cytoplasmic side of the plasma membrane. The web strengthens the cell surface, resists compression, and transmits force during cellular movement and shape changes. There's basically a dense web of thin actin filaments just deep to the plasma membrane, and this helps give the cell its shape and give it its strength and resist compression. Most microfilaments are involved in cell motility, which just means movement, or changes in cell shape. You can say that cells move when they get their actin together. Oh no, I wouldn't have run that if I saw that terrible joke ahead of time. Alright, for example, actin filaments interact with another protein, unconventional myosin, to generate contractile forces in a cell. So just how actin and myosin contract in muscles, the same thing happens in cells. I never learned this in high school and college A&P, so I always never understood how things could just move around in the cell without a muscle or anything like that. And they do it by having motor proteins that move along the cytoskeleton. That's how you move organelles around. And the cell itself, its structure actually has actin and myosin contractile units that can change its shape and make it move around. And that's how a macrophage can swim around and move around. The way actin and myosin work together, we'll learn a little bit more in the muscle chapter. Okay. This is basically what I just said, but this interaction also forms a cleavage fur that pinches one cell in two during division. Microfilaments attached to a cell adhesion molecules are responsible for the crawling movements of amoeboid motion and for the membrane changes that accompany endocytosis and exocytosis. Except in muscle cells where they are highly developed, stable, and long-lived, actin filaments are constantly breaking down and reforming. So any movement, whether it be of an organelle within the cell or of the cell itself, is accomplished in some capacity with the help of the cytoskeleton. Alright, intermediate filaments. These are tough and soluble protein fibers that resemble woven ropes. So these are bigger. They're the middle of the three. Made of twisted units of tetramer fibrils, they have a diameter between those of microfilaments and microtubules. Intermediate filaments are the most stable and permanent of the cytoskeletal elements and have high tensile strength. They attach to desmosomes, which are the structural cellular junctions that have actin anchors. And their main job is to act as internal guy wires to resist pulling forces exerted on the cell. Because the protein composition of intermediate filaments varies in different cell types, there are numerous names for the cytoskeletal elements. For example, they are called neurofilaments in nerve cells and keratin filaments in epithelial cells. Last part's not important for us. It's important for us, but not for our test. The one big difference between intermediate filaments and microfilaments is that intermediate filaments resist tensile forces and microfilaments resist compressive forces primarily. Also, intermediate filaments are bigger and are not made of actin. Okay, microtubules. These are the elements with the largest diameter. They're hollow tubes made of spherical protein subunits called tubulins. Most microtubules radiate from a small region of cytoplasm near the nucleus called the centrosome or cell center. We're going to talk about that a lot later. Microtubules are remarkably dynamic organelles, constantly growing out from the centrosome, disassembling, and then reassembling at the same or different sites. The stiff but bendable microtubules determine the overall shape of the cell, as well as the distribution of cellular organelles. So I made a big deal about the microfilaments. Organelles can be moved along across microtubules as well. Actually, primarily. So motor proteins will move the organelles, the vesicles, the mitochondria, using motor proteins called dynein and I can't remember. Dynein and the other one starts with a K. I don't think it's not in this book, but anyways, kinesin, maybe, I'm not sure. But these two motor proteins move organelles around in the cell along these microtubules. Oh, here we go. Kinesins and dyneins. Mitochondria, lysosomes, and secretory vesicles attach to the microtubules like ornaments hanging from tree branches. Tiny protein machines called motor proteins, kinesins, dyneins, and others continually move and reposition the organelles along the microtubules. So let me amend what I said a bit. So the myosin and actin microfilaments that lie just deep to the plasma membrane are responsible for cellular motion, whereas motor proteins and microtubules are responsible for organelle positioning and movement and vesicular trafficking, things like that. Movement within the cell. Motor proteins work by changing their shapes, powered by ATP. Some motor proteins appear to act like train engines, moving substances along on the microtubular railroad tracks. Others move hand over hand, someone like an orangutan, gripping, releasing, and then gripping again at a new site further along the microtubule. You guys have to look at this figure 3.24 on page 89. These motor proteins literally look like the lower half of our body, legs and a waist. And they walk along the microtubules. It's pretty crazy. And they just do that by having a conformational change in shape when they bind to an ATP molecule. So it's an active process. All right, that's it. Let's talk a bit about the centrosome. So the centrosome, or the center of the cell, it was mentioned earlier. The microtubules are anchored at one end in an inconspicuous region near the nucleus called the centrosome or cell center. The centrosome acts as a microtubule organizing center. It has a few distinguishing marks other than a granular-looking matrix that contains paired centrioles, which are these small barrel-shaped organelles oriented at right angles to each other. The centrosome matrix is best known for generating microtubules and organizing the mitotic spindle in cell division. Each centriole consists of a pinwheel array of 9 triplets of microtubules, each connected to the next by non-tubulin proteins and arranged to form a hollow tube. Centrioles also form the basis of cilia and flagella, which we're not going to talk about those. We're going to skip those for now. But so basically, there are these two little barrels oriented at right angles to each other that are the site of microtubule generation and microtubule generation. So microtubules radiate out from these, and make sure to look at the picture on page 89. And then they're disassembled and reassembled. And these centrioles are center stage when we talk about cellular division, and they make the mitotic spindle, which is basically these microtubules grow in a way that they actually push the two poles of the cell apart, and then the actin microfilaments pinch the cell in half at its center. So the cytoskeleton plays a big role, especially the centrioles and centrosome, in cellular division. We're going to skip cellular extensions, which are cilia and flagella on page 90, but feel free to check that out. It's interesting stuff. And we're going to go straight to... All right, guys, I accidentally just restarted the slide after recording on it for 20 minutes, so I'm going to start over, and I'm kind of glad I accidentally restarted it because I got a little complex, and I just want to keep it simple. So before I start, the nucleus. Okay, so we've probably all heard that the nucleus is the brain or control center of the cell, and that's true. But I think it kind of paints a weird picture, at least it did in my mind when I first heard that, of what the nucleus is and what it does. I pictured it as being like the brain, where it decides and dictates its function, the function of the cell, and that's not true. All the nucleus is just a membranous sphere, a lot like a cell really. It has a plasma membrane with a nucleolus inside of it and what looks like a cytoplasm, right? So it has that same general structure. And within this cytoplasm, which is called nucleoplasm, but we'll get to that later, there's chromatin. And this chromatin, however you want to say it, is basically the map for protein synthesis. So within this chromatin are nucleotide base pairs that code for protein. And that's it. That's all it does. It's just a container for our genetic material. Now, the way that it's able to dictate function of the cell is by reacting to certain hormones, which are fat-based, so they go through the plasma membrane of the cell, and then they go through the plasma membrane of the nucleus. These hormones and other triggers can cause the cell to make more of one type of protein or less of another. So it's not really a self-directed control center like our brain. It's more of a reactionary control center. It reacts to different triggers and hormones and things. And it's the proteins and enzymes within the nucleus that are reacting to these things. And it really, when it comes down to it, it's really just sort of a container for genetic material. Okay? All right, so let me read a bit here from the book. Actually, you know what? I don't think we're going to do that. Let's just talk about it a bit. So some cells are multinucleate, some cells are anucleate. So cells like muscle cells, for example, which are very long. If you think about a muscle cell in your quadriceps musculature, say your rectus femoris muscle, those muscle cells are maybe over a foot long. So there needs to be many, many nuclei because there's a lot of cytoplasmic mass that's going to be needing proteins to be built, developed, processed, sorted, things like that. The red blood cells are actually anucleate, mature red blood cells. They get rid of their nucleus in order to be smaller and to be able to fit through a capillary network and hold more oxygen and things. Because they don't have a nucleus, they can't replace proteins and enzymes as they degrade, which happens in about 3 or 4 months. So every 3 or 4 months, or these red blood cells can live for only about 3 or 4 months before they need to be replaced. And that happens in our red bone marrow. All right, there's 3 distinguishable regions of the nucleus. The first is the nuclear envelope, which is really the equivalent of the cell's plasma membrane, except that the nuclear envelope is much easier for things to pass through because it has these large nuclear pores. And these are aqueous pores that things can float in and out of, and primarily they're allowing for messenger RNA to enter and leave the nucleus. There's also nucleoli within the cell. We'll get into those in a second. And then there's chromatin. And I did want to read, I think on nucleoli and chromatin, we'll read through the book a little bit. So for nucleoli, these are the dark staining spherical bodies found within the nucleus where ribosome subunits are assembled. So this is where ribosomes are made. So ribosomes are what produce protein using the messenger RNA as a template, and we'll talk about that later. They are not membrane bound. Typically, there are 1 or 2 nucleoli per nucleus, but there may be more. Nucleoli are usually large and growing cells that are making large amounts of tissue proteins. Makes sense, right? Cells that need a lot of protein need a lot of ribosomes, so they need a lot of nucleoli. Nucleoli are associated with nucleolar organizer regions, which contain DNA that issues genetic instructions for synthesizing ribosomal RNA. As molecules of RNA are synthesized, they are combined with proteins to form the 2 kinds of ribosomal subunits. The proteins are manufactured on ribosomes in the cytoplasm and imported into the nucleus through these nuclear pores. Most of these subunits leave the nucleus through the nuclear pores and enter the cytoplasm, where they join to form functional ribosomes. So nucleoli, they produce ribosomes basically, is a really just simple way of looking at that. Chromatin. Okay, chromatin is very important. It looks like, when you look at the picture, it looks like it's just labeling what looks like it could be the cytoplasm of the nucleus. In the nucleus, we call this fluid interior the nucleoplasm, and the chromatin is just another component of that nucleoplasm. It's in the breakdowns here, it's DNA, histone, protein, and RNA chains within the cytoplasm. When the cell's going to divide, this chromatin condenses to form chromosomes. And I think we'll go to the book for this one too. So chromatin. Seen through a light microscope, chromatin appears as a fine, unevenly stained network. A special techniques reveal it as a symptom of bumpy threads weaving through the nucleoplasm. It's composed of approximately 30% DNA, 60% globular histone proteins, 10% RNA chains. Okay, so the functional unit of chromatin are nucleosomes. Okay, nucleosomes consist of flattened disc-shaped cores or clusters of 8 histone proteins connected like beads on a string by a DNA molecule. The DNA winds like a ribbon of Velcro twice around each nucleosome and continues on to the next cluster via linker proteins. So 8 of these histone proteins come together and then the DNA is wrapped around it. On and on and on and on, like these on a chain. This gives a way to condense the DNA, so it's not just free-floating and getting torn and broken in the nucleoplasm. Histones provide a physical means for packing a very long DNA molecules, some 2 meters worth per cell, in a compact, orderly way, but they also play an important role in gene regulation. I'm not going to go into this. Basically, depending on what type of compound is bound to the histone proteins, that can have an effect on the properties of the chromatin. So, for example, if, what are the examples they give in the book here? If phosphates are attached, it could mean the cell is preparing for programmed cell death. If acetyl is attached, that can expose certain regions of the chromatin to be transcribed to make proteins from, so specific genes need to be produced. Acetyls can be attached to that region, and other structures can be attached to deactivate certain genes. So it's not just a storage mechanism, it also can play a role in protein synthesis and cell function. All right. When a cell is preparing to divide, the chromatin threads coil and condense enormously to form short bar-like bodies called chromosomes. Chromosome compactness prevents the delicate chromatin strands from tangling and breaking during the movements that occur during cellular division. All right. So the nucleus, just a recap, plays a huge role in protein synthesis. And it does that by storing the DNA and being the site of much of the protein synthesis. It will transcribe the DNA to RNA messengers to go out to the cytoplasm. Ribosomes are produced here. Hormones, enzymes, and proteins determine which genes will be coded and which genes will be used to produce more proteins. So it's very important. And when cellular division takes place, it's going to be, its contents are going to need to divide so that both cells have identical DNA, identical genetic information. So let's look at that a bit. Let's look at the cell cycle, and then we'll get specifically into cellular division. All right, the cell cycle. So this is basically the life cycle of the cell, and this cycle occurs over and over, and one cycle is a generation. So we'll talk about that, what that means in a second. But basically, when we look at the cell cycle, there's really 2 basic phases, interphase and mitotic phase. So if you learned about mitosis before, that's when the cell divides, and that's what the illustration is showing, 2 cells being formed from one. Interphase is all of the time when the cell isn't dividing, or the time in between the mitotic phase. This is a little misleading because the cell is not just doing nothing during this time, it's growing, carrying out routine functions, and synthesizing DNA to prepare for mitosis. So let's go into interphase first and look at the subphases. So the first subphase is G1, or GAP1 subphase. The cell is metabolically active, synthesizing proteins rapidly and growing vigorously. And you can see this in figure 3.31, which is pretty much like the figure we have in the PowerPoint. This is the most variable phase in terms of length. Okay, so what this means is cells that divide often will have a very short G1 phase. Cells that rarely divide will have a very long G1 phase. Oh, and that's what it says here. Cells that divide rapidly, G1 typically lasts several minutes to hours only. But in those that divide slowly, G1 may last for days or even years. Cells that permanently cease dividing are said to be in the G0 phase. which isn't illustrated here, but that's when cells stop dividing, they're no longer in the cell cycle, they're just stuck in Z0. For most of G1, virtually no activities directly related to cellular division occur. However, as G1 ends, the centrioles start to replicate in preparation for cell division. So the centrioles, I don't know if you guys remember, but they were those little spindle structures that were oriented at right angles to each other. They start to replicate and prepare to perform all the movement and division that's going to take place during the cell, during the mitosis phase, mitotic phase. All right, S phase. DNA is replicated, ensuring that the two future cells being created will receive identical copies of the genetic material. New histones are made and assembled into chromatin. One thing is sure, without a proper S phase, there can be no correct mitotic phase. All right, so we're going to talk about DNA replication a lot later, but basically, S phase, DNA synthesis. It needs to make two copies of the DNA. In order to do that, you need to make new nucleosomes, which require histones proteins to be produced. And that's what it's talking about in the book. It says new histones are made and assembled into chromatin. So the first are assembled into nucleosomes, and then they're packaged as chromatin. They won't get turned into chromosomes until mitosis. Right now, it's just they're being packaged into chromatin, two identical sets of chromatin. All right, G2, GAP2 subphase. The final phase of interphase is brief. Enzymes and other proteins needed for division are synthesized and moved to their proper sites. By the end of G2, centriole replication, begun in G1, is complete. The cell is now ready to divide. Throughout S phase and G2 phase, the cell continues to grow and carries on with business as usual. So during all of interphase, the cell is still performing its functions that are required. Say if it's a beta cell in the pancreas, it's still producing the hormones it needs to produce, but it's also preparing for cellular division. Okay, let's talk about DNA replication. This isn't really a complex process, but you might need to go through it a couple times through the book to understand it. It's important to understand this, because when we look at protein synthesis, I want you to see the similarities and the differences between the two, between DNA replication and protein synthesis. They're very similar. Right. DNA replication. Before a cell can divide, its DNA must be replicated exactly, so that identical copies of the cell's genes can be passed to each of its offspring. During the S phase, replication begins simultaneously on several chromatin threads and continues until all the DNA has been replicated. Human DNA molecules are very long. Replication of a DNA molecule begins at several origins of replication along its length. So it doesn't just start at one end of the DNA and continue the whole thing. It starts at several different working sites. It begins at several origins of replication along its length that have a specific nucleotide sequence, a strategy that greatly increases the speed of replication. Okay, so let me back up. We didn't talk a whole lot about the structure of DNA. I'm not sure that was in this chapter. I think it might just be assumed that you've gotten that in the past, which I'm sure you have. But just a refresher, there's 4 nucleotide bases. Okay, A, T, C, and G. Okay, I always forget those. I think it's adenosine, where are they? Yeah, I don't know. It's not important for me, at least for my class. A, T, C, and G. These are molecules that make up codes. Okay, so they form a code. So a code that is GAU might code for a protein called aspartic acid, an amino acid called aspartic acid. AGC might code for, I forget, serine. So these base pairs make up a code for DNA. What's interesting, I want to make sure I get this right. We'll come back to that. But what's interesting is that, let's back up. So before we get into that, ATCG, those are your base pairs. DNA is 2 strands. So one strand has a code and the other strand has a code, and those codes have to bond to each other. But A's don't bond with A's, and T's don't bind with T's, and C's don't bind with C's. So what you need is to create base pairs. And what you should do is look at figure what figure is this? Figure 3.32 in your Marriott book to see what I mean. And here's what the base pairs are. A is adenine, T is thymine, C is cytosine, and G is guanine. So A, adenine or adenine, binds with T. A and T bind together. C and G bind together. So if one strand has an AAA code, the other strand would have a TTT code, and they would bind together. Okay. All right. I hope that wasn't too confusing. Let's look at the four events that occur in replication. So step 1. Enzymes attach to origins of replication and separate the DNA strands so that replication bubbles form. So these in this case it's DNA polymerase is this enzyme, and it's looking for a start code. It's looking for a series of 3 things that are going to trigger it to start synthesizing the DNA. And let's get into that some more. So step 1 again. Enzymes attach to origins of replication, which is whatever this code is, and separate the DNA strands so that replication bubbles form. So it's ballooning the DNA out the 2 strands so there's room to work. At each end of the replication bubble is a Y-shaped area, a replication fork, where the helical parental DNA strands are being unwound. So step 1 is basically enzymatic unwinding, and that's what we have here under DNA replication. Step 2, once the bubbles are formed, the parental DNA strands are ready to serve as templates for making the complementary DNA strands from free DNA precursors dissolved in the nucleoplasm. So all the structures to make DNA are just floating around in the nucleoplasm. However, there is a problem here because the polymerase enzymes that synthesize DNA cannot start a new DNA strand from scratch. They can only add new molecules to a strand that already exists. The problem is solved by formation of a short, complementary RNA primer, about 10 bases long, by a primase enzyme. So earlier I told you DNA polymerase is looking for the code. Sorry, it's RNA primer. This RNA primer is looking for the code that says start coding here. It's about 10 bases long, and it binds to the strand. RNA is a little bit different than DNA. It has one different base pair. It uses a U instead of a T. So its U base binds to an A instead of a T binding to an A. I think that's uracil. So this DNA polymerase can only add bases. So it finds the RNA primer and starts adding bases to that. And again, you really need to look at picture 3-32 on page 98 along with this while you're listening to this. It'll be really helpful. Okay, step 3. Continuing from the primer, the enzyme DNA polymerase positions complementary nucleotides along the template strand and then covalently links them together. DNA polymerase works only in one direction. Consequently, the leading strand, is synthesized continuously once primed, following the movement of the replication fork. On the other strand, called the lagging strand, is constructed in segments in the opposite direction and requires that a primer initiate replication of each segment. And then here's step 4. Ligase enzymes splice the short segments of DNA together. Eventually, DNA polymerases replace the primers with DNA nucleotides. All right. So the point of that is the lagging strand. The lagging strand has a lot more work to do. The leading strand just goes ahead and transcribes from start to finish. The lagging strand is going to have a lot more RNA primers, and those will need to be replaced by DNA. Is all that's saying. That's a bit complex for my class. So I just want you to read through that. I think the main thing I want you to understand is these steps. So step 1 is that the DNA is unwound and a replication bubble forms. And then step 2 is RNA primers need to bind to the start sites where the gene starts and give the DNA polymerase something to latch onto, which is step 3. The DNA polymerase latches on to the RNA primers, and then it gathers all the necessary bases out of the nucleoplasm and starts linking them together and making a new strand. Step 4 is that ligands come in and replace the RNA with DNA, the RNA primer with DNA, and they package and link the DNA. That's DNA replication in a nutshell. Again, in lecture, we're really going to want to look at figure 3.32. Okay. So that's DNA replication before cell division can occur. If there's no new DNA synthesis, DNA replication, then you wouldn't have two identical cells after division, and you'd have a problem. So when we talk about cell division, we're talking about the mitotic phase. So interphase. So when we look at the cell cycle, interphase was this long phase that was all the time the cell wasn't dividing. So the time in the cell is carrying out its normal functions that are required of it. During mitosis, the cell is only concerned about dividing. So let's read about mitosis. Mitosis is a series of events that parcels out the replicated DNA of the mother cell to two daughter cells. Described as 4 phases: prophase, metaphase, anaphase, and telophase. Mitosis is actually a continuous process, with one phase merging smoothly into the next. Its duration varies according to cell type, but in human cells it typically lasts for about an hour or less. Focus on mitosis describes the phases of mitosis in detail. So the book really doesn't go through it a whole lot. We're going to dive into it, though, pretty big. We're going to go through each phase here in a second. But I just want to talk about cytokinesis first. This is the final part of M phase. Cytokinesis, or the division of the cytoplasm, begins during late anaphase and is completed after mitosis ends. A contractile ring made of actin filaments, so these are microfilaments. If you remember when we talked about microfilaments earlier, there's this microfilament web just under the plasma membrane. So that's what we're talking about. A contractile ring of actin filaments draws the plasma membrane inward to form a cleavage furrow. And we're going to see a really good picture of that. Over the center of the cell. The furrow deepens until it pinches the cytoplasmic mass into 2 parts, yielding 2 daughter cells. Each is smaller and has less cytoplasm than the mother cell, but is genetically identical to it. The daughter cells then enter the interphase portion of the life cycle until it is their turn to divide. So cytokinesis basically divides the cell into 2 new cells, which are now smaller, but are identical to the mother cell. This is an awesome figure, guys. I would suggest studying this quite a bit. It's figure 3.33. It's 2 pages, and it breaks down mitosis into all of its steps. That's where we're going to start here with prophase. All right, so prophase has 2 parts, early and late. I'm going to read early prophase here from this figure. Early prophase. The chromatin condenses, forming bar-like chromosomes. So boom, very first thing that happens is the chromosomes are formed, and that's really important. In the top figure here on the PowerPoint, you can see that it's starting to form. Each duplicated chromosome consists of 2 identical threads called sister chromatids. Okay, so DNA replication produced 2 copies of genetic material. When it condenses into a chromosome, there's 2 identical bars that are bound at the middle by a centromere. Okay. Later, when the chromatids separate, each will be a new chromosome. Okay. The chromatids are these identical bars, and when they're bound together, they're a chromosome. As the chromosomes appear, the nucleoli disappear, and the 2 centrosomes separate from one another. Okay, so these are, remember that the centrosomes duplicated earlier in G1, I believe. They are now, as you can see in the picture, they are now pushing away from each other. Okay, and there you see the formation of an early mitotic spindle. That'll be important. The centrosomes act as focal points for growth of a microtubule assembly called the mitotic spindle. As the microtubules lengthen, they propel the centrosomes toward opposite ends of the cell. So as these lengthen, they're pushing the centrosomes apart. So try not to confuse centrosomes, centrioles, chromosomes, and centromeres. All these are really similar sounding and spelled. Make sure you go through and figure them all out before the test. Centromeres are what binds the 2 chromatids together in the nucleus, forming a chromosome. Okay? 2 sister chromatids bound by a centromere is a chromosome. The centrosomes are these little microtubule barrels that you see in the picture with the little things radiating out from them, the microtubules radiating out from them, and they're oriented at right angles to each other. There's one for each daughter cell. Right now they're in the same cell, and the microtubules are growing, pushing the centrosomes apart, which will eventually start to push the cells apart. Okay? And this microtubular arrangement that's pushing the cells apart is called the mitotic spindle. Whew, okay, that's rough. Microtubular arrays called asters extend from the centrosome matrix. That's the last step of early prophase. And that's labeled. The asters are labeled there. All right, late prophase. The nuclear envelope breaks up, allowing the spindle to interact with the chromosomes. So the nuclear envelope disappears. That's the major event that occurs in late prophase, which this illustration is called prometaphase. We'll refer to it as late prophase. So that step 1, nuclear envelope disappears. Step 2, some of the growing spindle microtubules attach to kinetochores, special protein structures at each chromosome centromere. Such microtubules are called kinetochore microtubules. So do you see how some of these microtubules have attached to the centrosomes? Centromeres. Oh, now I'm confusing you. They've attached to the centromeres in the chromosome, which is the center of the chromosome. And a microtubule from each centrosome is attaching to a centromere. So the centrosomes are these yellow, in this arrangement, these yellow centriole arrangements on either side of the cell that have microtubules growing out of them. So the mitotic spindle is pushing the cell apart, and then these kinetochore microtubules are attached to either side of the chromosome. So as the cell expands outwards, these chromosomes are being pulled apart. That's what's important here, okay? And look at these pictures and read through the book if that doesn't make sense, or email me and ask me some questions, because that's important. Metaphase. Let's move on to metaphase. Oh, here, let me just read these things here. This, for late prophase, I just want to cover it one more time. The nuclear envelope dissolves. That's important. Spindle microtubules attach to kinetochores at each centromere. I wish you could see my mouse so I could point to that. Polar microtubules slide past each other, forcing the poles apart. So polar microtubules are forcing the cell apart, and the kinetochore microtubules are pulling the chromosomes apart. Okay, they're not so forceful that they're actually tearing the chromosomes apart, but they're pulling them apart, which is going to make them eventually line up along the equator of the cell, and that's what we're going to see next in metaphase. All right, guys. So in this slide here, we see metaphase. Now, if you look at the picture on the right, you'll see that the centrosomes are at the opposite poles of the cell. So their polar microtubules have been pushing them apart, which is creating two poles of the cell. And it's actually starting, it hasn't really started yet, but it's starting to stretch the cell apart. Okay. Also, there's kinetochore spindles attached to the centromeres of the chromosomes. All right. And because they're pulling on both sides of the chromosome, it's causing the chromosomes to align along the equator of the cell. Oh, and this is all listed over here on the right. That's here we go. Causing the chromosomes to align along the equator of the spindle, and this is called the metaphase plate. Okay. Now, this is when enzymatic separation of the chromatids begin. So the chromosomes are going to start to be enzymatically cut in half at the centrosome. Sorry, not the centrosome. At the centromere. It looks like I might have labeled this wrong. If you look here, I put centromeres are at the opposite poles of the cell. That should be centrosomes are at the opposite poles of the cell. Sorry about that. The centromeres are what's holding the sister chromatids together. So the centrosomes are opposite parts of the cell, opposite poles of the cell. The chromosomes are aligned along the equator of the spindle, which is called the metaphase plate, and enzymatic separation of the chromatids begins. Okay, so let's go ahead and move on to the next slide. Anaphase. I'm not sure why I changed the title from mitosis to anaphase. Just ignore it. This is still a phase of mitosis. again, it goes prophase early, prophase late, metaphase, which you just went over, then anaphase, which we're about to talk about now, and lastly, telophase. So anaphase is the shortest phase of mitosis. It begins abruptly as the centromeres of the chromosome split simultaneously. So if you remember in the last phase, metaphase, the chromatids began to become enzymatically detached from each other. But remember that the kinetochore microtubules, which are attached to the centrosomes at either pole of the cell, are also attached to the centromeres at the chromosomes. So when the chromosomes are enzymatically detached, they're anchored. The two chromatids, the sister chromatids, are anchored to opposite poles of the cell. Okay. Each chromatid now becomes a chromosome in its own right. So once it splits, it goes from being two chromatids bound together to two chromosomes that have been torn apart. The kinetochore microtubules, moved along by motoproteins in the kinetochores, gradually pull each chromosome towards the pole it faces. At the same time, the polar microtubules slide past each other, lengthen, and push the two poles of the cell apart. So the cell is starting to be stretched apart, and simultaneously the chromosomes are being reeled in to opposite poles of the cell. The moving chromosomes look V-shaped. Why do you think that is? That just kind of makes common sense, right? Let's read it from the book. The centromeres lead the way, and the chromosome arms dangle behind them. So they're literally being pulled towards the pole. So they're being pulled by the centromere, so it causes them to form a V as they're being dragged along through the cytoplasm. Moving and separating the chromosomes is helped by the fact that the chromosomes are short, compact bodies. Diffuse threads of chromatin will trail, tangle, and break, resulting in imprecise parceling out to the daughter cells. This is just saying why it's important that they're in bundled chromosomes. All right, so let's just do a quick overview of anaphase, and then we'll move on to telophase. All right, so anaphase, it's the shortest phase in mitosis. It begins with chromatid separation, simultaneous chromatid separation. Once that happens, these sister chromatids become chromosomes. All right, and they become two pairs of daughter chromosomes. Sorry, not two pairs. Two daughter chromosomes, which are identical. Motor proteins within the kinetochores pull chromosomes towards the opposite poles. Polar microtubules continue to expand, pushing the poles farther apart. And that's why the cell begins to elongate. Telophase. Telophase begins and the chromosomal movement stops. So as soon as the motor proteins and the kinetochores are done reeling the chromosomes in, telophase begins. It resembles prophase in reverse. Chromosomes unravel and revert to chromatin. Because you remembered early prophase, the chromatin condensed and formed chromosomes. This is now the reverse. It's going from a chromosome to chromatin. It's dissolving. A new nuclear envelope forms, and nucleoli form. The mitotic spindle disappears. Mitosis ends, and the cell is now binucleate. It's still attached, so it's two identical cells that are still attached in the middle. But the cleavage furrow is formed. You can start to see where it's starting to pinch off. Okay? That leads into our next step, cytokinesis. This is when that actin ring we were talking about forms and constricts until the cell is pinched in two. This begins during late anaphase and continues beyond telophase. So the cleavage furrow started forming in late anaphase, and it's not completely done pinching and dividing the cells until beyond telophase, until after mitosis is complete. And that's mitosis. There's also meiosis. We're not going to talk about that here. We'll talk about that in another section of the book. But let's move on to protein synthesis. This is kind of the big one here. There's a bit to it. This is why DNA exists right here. It exists to function as code for proteins and enzymes in our cells and in our body. So I'm just going to go ahead and read through this first intro paragraph of protein synthesis, and we'll talk about it a little bit. In addition to directing its own replication, DNA serves as a master blueprint for protein synthesis. Although cells also make lipids and carbohydrates, DNA does not dictate their structure. Historically, DNA is said to specify only the structure of protein molecules, including the enzymes that catalyze the synthesis of all classes of biological molecules. So DNA doesn't code for carbohydrates or for sugars, but it produces the proteins that modify them, that pump them in and out of the cell, and other things. Much of the metabolic machinery of the cell is concerned in some way with protein synthesis. This is not surprising, seeing that structural proteins constitute most of the dry cell material, and functional proteins direct almost all other cellular activities. Essentially, cells are miniature protein factories that synthesize a huge variety of proteins that determine the chemical and physical nature of cells, and therefore the whole body. Okay, that's way back in the first slide from cell theory, when we said that when you talk about the property of cell, you're talking about the property of the body. Recall from chapter 2 that proteins are composed of polypeptide chains, which in turn are made up of amino acids. For purposes of this discussion, we define a gene as a segment of a DNA molecule that carries instructions for creating one polypeptide chain. Supposedly, we humans have an estimated 25,000 protein-coding genes. Note, however, that some genes specify the structure of certain varieties of RNA as their final product, and not protein. Okay, that was a whole lot of words to say DNA is the blueprint for protein synthesis. And it's also the blueprint for RNA, which plays a big role in protein synthesis, and we'll see that here shortly. Okay, let's keep talking about DNA though. The four nucleotide bases, A, G, T, and C, are the letters used in the genetic alphabet, and the information of DNA is found in the sequence of these bases. Each sequence of 3 bases is called a triplet and can be thought of as a word that specifies a particular amino acid. For example, the triplet AAA calls for the amino acid phenylalanine, and CCT calls for glycine. The sequence of triplets in each gene forms a sentence that tells exactly how a particular polypeptide is to be made. It specifies the number, kinds, and order of amino acids needed to build a particular polypeptide. All right, so this is just here saying that these nucleotide bases code for certain amino acids. Every sequence of 3 nucleotide bases is a code for a certain type of amino acid. The order that these amino acids are coded for in the DNA determines the structure of the protein. So if you have, 300 A's in a row, you're going to have a protein made out of 100 phenylalanine, because AAA is a code for phenylalanine. All right. Same thing if you have a CCT, CCT, CCT over and over again, you're going to have a glycine bound to a glycine bound to a glycine. Variations in the arrangement of A, T, C, and G allow our cells to make all the different kinds of proteins needed. Even a small gene has an estimated 210 base pairs in sequence. The ratio between DNA bases in the gene and amino acids in the polypeptide is 3:1. That makes sense, right? If you have 300 base pairs of a gene, you'll have an 100 amino acid sequence. So you would expect the polypeptide specified by a 210 base pair gene to contain 70 amino acids. That's out of the book. Most genes of higher organisms contain exons. So we're down here at exons and introns. Exons are amino acids specifying informational sequences. Exons are often separated by introns. So exons basically are genes. Introns are non-coding, often repetitive segments that range from 60 to 100,000 base pairs, once considered a type of junk DNA. So this is DNA that doesn't code for anything. Intron DNA is believed to serve as a reservoir or scrapyard of ready-to-use DNA segments for genome evolution, as well as a rich source of small RNA molecules. The rest of the DNA, indeed most of it, is essentially dark matter, whose function is still a mystery. It is in these regions that pseudogenes, false genes, are found. Pseudogenes are genetic fossils that are the remains of damaged genes. They look like real genes but have deficits that render them functionless. So exons are where the map for the proteins are. The map for the amino acid sequencing is. They're essentially genes, exons. Introns are all these base pairs in between these genes that don't necessarily code for anything. And here the book is saying maybe it's a scrapyard to make new genes, or maybe it's maybe they're remnants of old genes that have problems now that we're not using anymore. Because this DNA has been passed on through the generations, so it makes sense that there will be damaged genes in a place to make new genes. The role of RNA, right? That's the next section I'll be reading. By itself, DNA is like a CD recording. The information it contains cannot be used without a decoding mechanism, a CD player in this analogy. Furthermore, most polypeptides are manufactured at ribosomes in the cytoplasm, but in interphase cells, DNA never leaves the nucleus. Okay? So, in interphase, which is everything when your cell is not dividing, interphase is the time the cell is not dividing. DNA can't leave the nucleus. It's bound there. The only time DNA leaves the nucleus is during mitosis when the nuclear membrane dissolves. Okay, so we need something to go into the nucleus and make a recording of the DNA, and then where it can be brought back out into the cytoplasm. DNA requires not only a decoder but a messenger as well. So that's basically what I was just saying. The decoding and messenger functions are carried out by RNA, the second type of nucleic acid. All right, so DNA is deoxyribose nucleic acid. RNA is ribose nucleic acid. As you learned in Chapter 2, which we skipped, RNA differs from DNA. RNA is a single-stranded structure. DNA is a double-stranded structure. RNA is single-stranded, and it has a sugar ribose instead of deoxyribose, and the base uracil instead of thymine. 3 forms of RNA typically act together to carry out DNA's instructions for polypeptide synthesis. And the 3 types are messenger RNA. Let's go to the next slide. The 3 types are messenger RNA, ribosomal RNA, and transfer RNA, or transport RNA. Messenger RNA. This is a relatively long nucleotide strand resembling half DNA molecules, 1 of 2 strands of a DNA molecule coding for the protein structure. We'll talk about that just now. mRNA carries a transcript of the code to the cytoplasm where protein synthesis occurs. Okay, so we're going to get into transcription and translation later, and those are going to involve all of these. But basically, messenger RNA or mRNA. Sorry, I got a phone call. I don't quite remember where I was, but messenger RNA basically goes into the nucleus, and let me start over. In the nucleus, DNA is unwound, and messenger RNA is formed. It makes a transcript. It makes a matching copy of one of the strands of DNA. Then that comes out into the cytoplasm where it can be turned into protein. Okay. Ribosomal RNA is the building block of ribosomes. It's important in protein synthesis because it allows the ribosomes to bind to the RNA, and we get to transcribe. Transfer RNA, this is really important. This transports cytoplasmic amino acids to the ribosomes. So the ribosomes they bind to the messenger RNA and they're floating around in the cytoplasm. Then the transfer RNA brings in what's called codons, which we'll talk in a second, but brings in amino acids to be fused to this messenger RNA, and then that forms the amino acid. So DNA is the map, and RNA is what's reading the map and what's actually making the protein. All right, so there's two big steps to protein synthesis: transcription and translation. Let's jump right into transcription, and there's a picture here of transcription on this slide. A transcription, and this is from the book. A transcriptionist converts a message from a recording or shorthand notes into a written copy. In other words, information is transferred from one format to another. In cells, transcription transfers information from a DNA-based sequence to the complementary base sequence of an mRNA molecule. The form is different, but the same information is being conveyed. So in ACG, will now become a UGC. Okay, so it's a different sequence of base pairs because they're opposite bases to the DNA. So if it's an A on a DNA, it's a U on an RNA. If it's a G on a DNA, it's a C on an RNA, right? So it's a different code, but it still codes for the same proteins. Okay, it's still accomplishing the same thing. Same information, but a different format. Once the mRNA molecule is made, it detaches and leaves the nucleus via a nuclear pore and heads for the protein synthesis machinery, the ribosome. Transcription cannot begin until gene-activating chemicals called transcription factors stimulate histones at the site to be of gene transcription to loosen. So transcription factors have to cause the DNA to unwind, and these transcription factors are activated by a number of things, hormones, steroids, whatever. The transcription factors then bind to the promoter. The promoter is a special DNA sequence that contains the start point, the beginning of the gene to be transcribed. It specifies where mRNA synthesis starts and which DNA is going to serve as the template strand. Okay. So the start compound that it's looking for. Oh, sorry, no, that's something else. So it's just the promoter is just a special DNA sequence that contains the start point. Okay. So RNA polymerase, after this is unwound and this bubble forms, RNA comes in and moves along and is looking for the promoter, which is a special type of base sequence. Okay. Once it finds a promoter, it starts to put down new bases and form the mRNA strand. The template strand is the strand that is being, how do I say this? Okay, so if you look at the image, you see that the helix is being pulled apart. Okay? The template strand is the strand that's being transcribed. Okay? The coding strand is the other strand. That is the strand that the RNA is going to match. Remember? Because the template strand binds to the coding strand and is opposite. Okay? They're detached, and essentially a new coding strand is made out of mRNA. So the coding strand of DNA and the messenger RNA strand will actually match, with the exception that the T's on the coding DNA strand will be U's on the mRNA strand. Okay, the uncoiled DNA strand, not used as the template, is called the coding strand because it has the same coded sequence of the mRNA to be built, except for the U in mRNA in place of T in DNA. So this is just what I said, but out of the book. The transcription factors also help position RNA polymerase, the enzyme that oversees the synthesis of mRNA, correctly at the promoter. Once these preparations are made, RNA polymerase can initiate transcription. Transcription involves three basic phases: initiation, elongation, and termination. Let's go through those right now, okay? Once properly positioned, RNA polymerase pulls apart the strands of the DNA double helix so transcription can begin at the start point in the promoter. Step 2, elongation. Using incoming RNA nucleotides as substrate, the RNA polymerase attaches them with complementary DNA bases on the template strand and then links them together. As RNA polymerase elongates the mRNA strands of the mRNA strand one base at a time. Oh my goodness. As RNA polymerase elongates the mRNA strand one base at a time, it unwinds the DNA helix in front of it and rewinds the helix behind it. At any given moment, 16 to 18 pairs of DNA, base pairs of DNA, are unwound, and the most recently made mRNA is still hydrogen bonded to the template DNA. This small region called the DNA-RNA hybrid is up to 12 base pairs long. I wish I could laser point right now, but if you look at this image, you have this big blue blob, right? That's RNA polymerase. As it moves down the strand, it unwinds it. Okay? And then it starts to attach nucleotide pairs, or nucleotide bases, to the template strand. Okay? And it gets about 10 to 12 base pairs long, or up to 12 base pairs long, where the RNA and the DNA are actually attached. And you can see that where the red and the blue are attached there. Then it starts to detach as it moves on. It starts to detach the lagging RNA, the lagging end of the RNA, and rewind the DNA helix as it moves on. So in this case, it's moving to the right. So you can see in the image it says DNA is unwound on the right, and DNA is rewound on the left. So it's unwinding and rewinding as it moves along. And it's adding nucleotides at the DNA RNA hybrid, which is up to 12 base pairs long. And that is where the red and blue are intertwined. And then it detaches and an mRNA strand is created and starts to trail the RNA polymerase as it moves along. Okay, termination. This is step 3. When the polymerase reaches a special base sequence called a termination signal, transcription ends and the newly formed mRNA separates from the DNA template. So now we have an RNA template that can leave the nucleus that matches the coding strand, which codes for a particular protein perfectly. Okay, it matches the DNA code perfectly. It has U's instead of T's, but it will still code for the right amino acids. All right, so that was it for transcription. Again, transcription is just a creation of this mRNA strand that codes for a protein. Now it needs to be translated from mRNA into protein. That's what translation is. But first, let's finish off transcription. I kind of skipped over something, and this is the processing of mRNA. So mRNA is a product of transcription. It just needs to be processed a little bit. So before translation can begin, editing and further processing are needed to clean up the mRNA transcript. Recall that mammalian DNA, like ours, has coding regions exons separated by non-protein coding regions introns. Because the DNA is transcribed sequentially, the mRNA initially made, called pre-mRNA, or primary transcript, is still littered with introns. Before the newly formed RNA can be used as a messenger, it must be processed or edited. That is, sections corresponding to introns must be removed. Large RNA complexes called spliceosomes snip out the introns and splice together the remaining exon-coded sections in the order in which they occurred in the DNA, producing functional mRNA. Although many introns degrade naturally, some contain active segments, such as microRNAs, that can function to control, interfere with, or silence other genes. Additionally, a number of specific RNA binding proteins called mRNA complex proteins must become associated with the edited mRNA. These mRNA complex proteins guide the export of mRNA from the nucleus, determine its location, translation, and stability, and check it for premature termination codons. All right, so basically, after we get our pre-mRNA transcribed, it needs to be edited. We need to get rid of the introns, which are regions of basically garbage RNA. We get rid of them. We link all the exons, which are the coding regions of the gene, together. And then that is exported from the nucleus out into the cytoplasm. There, it's going to bind with the ribosome, and translation can begin. All right, I'm going to read the translation section now. The translator makes a message in one language, takes a message in one language, and restates it in another. In the translation step of protein synthesis, the language of nucleic acids, which is base sequence, is translated into the language of proteins, which is an amino acid sequence. Okay, genetic code. The rules by which the base sequence of a gene is translated into amino acid sequence are called the genetic code. For each triplet or 3-base sequence of DNA, the corresponding 3-base sequence of mRNA is called a codon. Okay. So each codon is 3 base pairs of functional mRNA. That's called a codon. It's called a codon because it codes for a specific amino acid. And we can see that in this little graph here. We see all these 3 sequence of letters, UUU, UUC, UUA, UUG. Those all code for a specific type of protein. So UCU codes for serine. GGU on the bottom right codes for glycine. They all code for different amino acids. So by putting these codons in a certain order, we develop a chain of different amino acids. Since there are 4 kinds of RNA or DNA nucleotides, there are 4 cubed or 64 possible codons. Three of these 64 codons are stop signs that call for termination of polypeptide synthesis. All of the rest code for amino acids. That means there's 61 different amino acids that can be assembled in different orders. Actually, sorry, that's not true. There's less than that, but there's 61 codons that code for different amino acids. How many are there? 1, 2, 3, 4, 5, 6. All right, so it looks like there's about 22 or 23 amino acids that can be arranged in different orders to make different proteins. And those are all coded for by the 61 different amino acid coding codons. Because there are, that's the next sentence. Because there are only about 20 amino acids, some are specified by more than one codon. This redundancy in the genetic code helps protect against problems due to transcription and translation errors. Figure 3.36 shows the genetic code and a complete codon list. So check that figure out in the book. It's a lot like this one here. All right, so that's what mRNA is, functional mRNA. It's transcribed in the nucleus, it's edited, then it comes out and it contains sequences of codons that contain amino acids. Let's look at the role of tRNA in translation. Translation involves the mRNAs, tRNAs, and rRNAs mentioned above. Before we get into the actual details of the process, let's look at how tRNAs are so well suited for their roles in translation. Shaped like a handheld drill, tRNA is well suited to its dual function of binding to both an amino acid and an mRNA codon. The amino acid picked up from the cytoplasm is bound to one end of the tRNA at a region called the stem. At the other end, its head is its anticodon, a 3-base sequence complementary to the mRNA codon calling for the amino acid carried by that particular tRNA. Okay, so say we have a UUU codon. There's a tRNA with a TTT codon, anticodon, is going to bind to that codon. So a TTT will bind to a UUU codon. Now, because it's binding to a UUU codon, it will be carrying a phenyline, a PHE amino acid with it. Okay? You can see that in this picture that if we look on the right side, the bottom right picture, you see there's 3 labels on the right-hand side of the image: amino acid, tRNA, and anticodon. Okay? So the anticodon binds to the appropriate codon and carries the amino acid that's specified by the codon. All right. Hopefully that makes sense. We'll talk about it in lecture more, and I really suggest looking at figure 3.37 in your textbook. That's on page 106 and 107. It's really helpful. So go ahead and read through that and look at the pictures. Pictures help me a lot. So. Okay, so let's start over with anticodon. At the other end, its head is this anticodon, a 3-base sequence complementary to the mRNA codon calling for the amino acid carried by that particular tRNA. Because anticodons form hydrogen bonds with complementary codons, tRNA is the link between the language of nucleic acids and the language of proteins. For example, if the mRNA codon is AUA, which specifies isoleucine, the tRNAs carrying isoleucine will have the anticodon UAU. UAU binds to AUA, makes sense? Which can bind to the AUA codons. There are approximately 40 types of tRNA, each capable of binding with a specific amino acid. The attachment process is controlled by amino acid tRNA synthase enzyme and is activated by ATP. Once this amino acid is loaded, the tRNA, now called an amino acetyl tRNA because of its amino acid cargo, migrates to the ribosome, where its amino acid is maneuvered into the proper position, as specified by the mRNA codons described below. The ribosome is more than just a passive attachment site for mRNA and tRNA. Like a vice, the ribosome holds the tRNA and mRNA close together to coordinate the coupling of codons and anticodons. It positions the next incoming amino acid for addition to the growing polypeptide chain. To do its job, the ribosome has a binding site for mRNA and 3 binding sites for tRNA. an A amino acyl site for an incoming amino acyl tRNA, a P peptidal site for the tRNA holding the growing polypeptide chain, and an E exit site for an outgoing tRNA. This is illustrated on figure 3.37 on page 106 to 107. So make sure you look at that, because without a picture, all those words are insane. And this picture I have here isn't that great. I really couldn't find one I like perfectly online. But let's go through the 3 steps of translation. And after that, we're going to be done, and we'll talk about the study guide. Actually, we'll talk about the study guide in class. All right, step 1, initiation. A small ribosomal subunit binds to a special initiator tRNA. Okay, so the ribosome binds to an initiator tRNA, and then to the new mRNA to be coded. So it starts right at the very first base that's going to be decoded. When the initiator tRNA is still in tow, the small ribosomal subunit scans along the mRNA until it encounters the start codon. The start codon is the first AUG triplet, or the first AUG codon it meets. So it scans along the mRNA until it gets to an AUG, and then boom, translation starts. When the initiator tRNA's UAC, which is the opposite of AUG, anticodon recognizes and binds to the start codon, a large ribosomal subunit unites with the small one, forming a functional ribosome. Okay, so that's a lot of stuff happening. So basically, the small subunit of the ribosome binds to a tRNA molecule, an initiator tRNA molecule, and then it scans along the mRNA looking for an AUG. Because the initiator tRNA has a UAC anticodon, it binds to the AUG, because UAC binds to AUG. Once that happens, a large ribosomal unit attaches, boom, sandwiches the mRNA in between the 2 ribosomal subunits, becoming a functional ribosome. At the end of this phase, the mRNA is firmly positioned in the groove between the ribosomal subunits. The initiator is sitting in the P site. The A site is vacant, ready for the next amino acyl tRNA to deliver its cargo. Remember, there's 3 sites in a ribosome. A P site is the first one, an A site, and then an E site. The P site right now is filled with tRNA. The A site is vacant, ready for the next tRNA to deliver its amino acid. The next phase, elongation of the polypeptide, now begins. So initiation ends with a ribosome, a full functional ribosome bound to mRNA with only the P site occupied. Elongation, step 2. During the three-step cycle of elongation, the mRNA is moved through the ribosome in one direction. And one amino acid at a time is added to the growing polypeptide. Okay? So this is broken down into 2A, 2B, and this is weird, 2A, 2B, and 2C. Okay. 2A, codon recognition. The incoming amino acetyl tRNA binds to a complementary codon in the A site of a ribosome. Okay, so the A site now becomes occupied. Step 2B, peptide bond formation. Once the accuracy of the codon-anticodon binding is checked, an enzymatic component in a large ribosomal subunit catalyzes peptide bond formation between the amino acid of the tRNA in the P site to that of the tRNA in the A site. So remember, every piece of tRNA has an amino acid bound to it. As new tRNAs come into the ribosome, their amino acid that's attached starts being bound together and making a chain, which is illustrated in this picture on the bottom right. Step 2C, this step translocates or moves the tRNA in the A site to the P site. The unloaded vacant tRNA is transferred to the E site, from which it is released and ready to be recharged with an amino acid from the cytoplasm. Okay, so these sites are the E site, something is released, and then it moves down, and then something moves into the P site. And then it moves on like this, down and down and down. And this is all illustrated in the picture on the next page. This orderly musical chairs process continues. The peptidyl tRNAs transfer their polypeptide cargo to the aminoacetyl tRNAs, and then the P site to E site and A site to P site movements of the tRNAs occur. As the ribosome chugs along the mRNA track and the mRNA is progressively read, its initial portion passes through the ribosome and may become attached successively to several other ribosomes, all reading the several message simultaneously and sequentially. This multiple ribosome mRNA complex, a polyribosome, efficiently produces multiple copies of the same protein. So basically what this is saying is that one strand of mRNA can be moving through multiple ribosomes at one time. So one strand of mRNA can be producing several protein chains at once. Okay. Step 3, termination. The mRNA strand continues to be read sequentially until its last codon, the stop codon, which is either UGA, UAA, or UAG, enters the A site. The stop codon is the period at the end of the mRNA sentence. It tells the ribosome that translation of that mRNA is finished. As a result, a protein release factor binds to the stop codon at the A site and directs the addition of water instead of amino acid to the polypeptide chain. This I can never say these words. This hydrolyzes breaks the bond between the polypeptide and the tRNA in the P site. The completed polypeptide chain is then released from the ribosome, and the ribosome separates into 2 subunits. The released protein may undergo processing before it folds into its complex 3D structure and floats off, ready to work. When the message of the mRNA that directed its formation is no longer needed, it is degraded. There is a lot to that, you guys. So protein synthesis it's essentially what our cells do. It's their purpose, is to make protein. They're little protein factories. I really don't expect you to understand that completely because you guys only have a lot of good images. So when you listen to this, make sure you have your book with you, and maybe listen to it again and go through the steps on the on page 106 and 107 as I talk. Go ahead and follow the steps through it. It has some really good images on there. Okay, so that's it. I was hoping an hour and a half or so, but I basically took up how long our class is going to be, 2 hours and 50 minutes. These are some images here on this slide that you should just check out, 339 and 347. Sorry, 3.39 and 3.4. They're just additional images to help you understand, and there's a really good image. 3.39 shows how one mRNA strand can move through multiple ribosomes. In this case, it's the rough endoplasmic reticulum. You can see how the rough endoplasmic reticulum goes about packaging and producing proteins. It's pretty cool. So this next slide, I'm going to give you guys a study guide in class. I'm hoping that's helpful. This is a lot of material, so I know that's going to be helpful to have a study guide. All right, so I went back and listened to this, and man, I'm clicking my pen the whole time, and my dogs are playing in the background. I hope that doesn't throw you guys off. Anyways, I hope this was helpful. I think the next webinars will continue to get better. This was just our first shot, and let me know if you like it, and we'll keep doing it. Okay. All right. Good luck.